Okay, so let's be crazy and start in order. Number one. Two populations of a species of squirrel are geographically isolated from each other. Okay, although they have the same population density, one population is significantly larger in number than the other. A new bacteria disease, a new environmental factor is easily spread and extremely virulent, which means it's deadly, affects both populations at the same time. Which of the following is the best prediction of how the new disease will affect the two populations? Okay, well, what we do know is that if there are two populations of the same species, that means they are, of course, able to breed amongst each other. There's no reproductive isolation. And the key here, party people, is that there is one population that is significantly larger. And if they are geographically isolated, there means there is no gene flow. Now, if there's no gene flow between the two populations, what could happen is the smaller population could have a very, very different percentages of alleles and types of alleles that exist. Now, why do I say that? Now, what's an allele? An allele is an expression of a gene. And we think of alleles sometimes as many different forms or expressions of the same gene. For instance, hair color, eye color, flower color. You have a gene for the color of the flower, but you can have many different types of flowers. But the bottom line here is that they're separated and there's no gene flow. So with a smaller population, okay, what you could have is something called genetic drift, where the types of alleles and the percentage of alleles can be far different than a larger population because by chance events, okay, a certain number of individuals with a certain number of alleles could die off and not mate. When you have small populations, okay, very small populations especially, you can have this genetic drift where the types and percentage of alleles can change extremely fast. In fact, in some cases you may lose certain alleles by just the chance events of the mating or lack of mating or just the death of a certain different individuals, or just a chance of how gametes come together to make zygotes. Okay, when you have larger population, again, great example of that is flipping a coin. You know, a coin can be heads or tails, and we know that if we flip it 4 or 5 times, there's a good chance we can have 4 or 5 heads in a row. But if we flip it 1,000 times, okay, those percentages will work out almost exactly 50 and 50. So back to this, we have no genetic flow. There's no genes flowing back and from, and we should know that the smaller population, because of the chance events that occur and affect smaller populations more, genetic drift, we're probably going to have smaller populations have less variability, genetic variability, because of the loss of some alleles that could occur. Smaller the population, the lower, okay, the genetic variability. All right, and probably a better term is genetic diversity. We have many more different types of alleles and many different forms or of the same gene available, they're better able to what? Combat any future environmental conditions. So if we've got a bacteria disease, okay, probably most definitely the larger population with more alleles present, okay, would be able to probably have an allele that is resistant to that bacteria disease, whereas the smaller population with the lower diversity probably does not have that mutant in its population, and therefore the survival of the smaller population should be lowered. So the answer to our question, the two populations will equally affected? Most definitely not. Again, this is all chance. It'll be the smaller population who will be affected more. The larger population will be less affected by disease than will the smaller population because the mutation rate of the larger population. Mutation rate is constant for all organisms, so it's not going to be any different because of the size of the population. Although the first part is right, the second part is wrong. That's what makes it a very nice distractor. C, the smaller population will be more affected. Yes, that part is right. Don't just stop reading, as we've seen before. Okay, first part is right because the smaller population has less genetic variation, and that is by far our best answer. Okay, all right, so the smaller population will be less affected, the smaller population will be more affected. So again, C is the best choice. Moving forward, number 2. Number 2, mitochondria are found in most eukaryotic cells and contain their own DNA and ribosomes that are similar to those typical of many prokaryotic cells. Which of the following best statements is justified by these observations? Well, party people, okay, if they are typical of many prokaryote cells, this question is really about endosymbiosis, all right? And endosymbiosis is the idea that an ancestral cell somehow engulfed, okay, a prokaryote, and that prokaryote eventually became the organelle mitochondria. How do we know? Well, if this is a living organism that was engulfed, it will have, like all living things, its own DNA. And because mitochondria, like we've talked about, has their own plasmids, okay, and is very efficient in making energy because the nucleus of the cell does not necessarily control these genes, okay, although there are genes that interact between the nucleus of the cell and the mitochondria makes most of its own enzymes and proteins that do its own energy, help it with the energy efficiency. Any case, so this is really speaking to the idea of endosymbiosis, and of course we have this idea with chloroplasts as well. So looking at the choices, the mitochondrion is the only location in which eukaryotic cells can synthesize ATP. That's wrong. We can make ATP without mitochondria. Glycolysis, which is a much older energy system, is anaerobic. B, an ancestral cell most likely engulfed an aerobic prokaryote in a relationship that was proved beneficial for both cells. Boy, that's the answer. No really reason to go on, but hey, mitochondrial membranes provide abundant surface area for reactions because of the infolding called cristae. That is true, absolutely true, that cristae increases the surface area when you draw those folds inside the mitochondria. And it's in these membranes right here where we have the phosphorylation, the oxidative phosphorylation, and of course that's where we have the ATP synthase. So all the action is on that membrane, and of course that membrane is folded to increase the surface area so more of it happens. So 2 is C. All right. And then D, the mitochondrion plays a role in respiration, but it also stores the cell's extra messenger RNA. And then number D, as far as I understand it, doesn't make any sense to me. Mitochondrion plays a role in respiration. Absolutely correct. But also stores the cell's extra messenger RNA. Why would we want to store any extra messenger RNA after messenger RNA is done in its translation? Is basically cut up so that it can be reused. So this doesn't make much sense to me. So the answer, okay, is that even though C is the correct answer, it's not the correct for the answer. So B, an ancestral cell most likely engulfed an aerobic prokaryote. So this is what we just talked about in terms of they were after the endosymbiotic relationship. All right, so that's number 2 is B, and going on to number 3. So mutation question, number 3, sickle cell anemia. That's where we have our normal red blood cells have this concave kind of disc, and then sickle cell, because of the shape of the protein, we have these very thin spiny type of red blood cells that can get caught up in capillaries and form clots. Sickle cell anemia is associated with a mutation in the gene encoding the beta subunit of hemoglobin, the protein, okay, by which gives the red blood cell its shape and its ability to gather oxygen. that results in a change from glutamic acid to valine. So we have a substitutional type of a mutation. So at position 6, all of the amino acids are identical to the normal. That's key here, party people, that in this mutation, all the other acids are identical. Why is that key? That's key because if they were not identical, that would mean that we have a frame shift mutation, whereas, if we have a triplet codon, if we were to take one, it's called an insertion or a deletion, we would move the reading frame back forward or front forward. So we can get rid of an insertion or a deletion, okay? And this would cause a frame shift. And a frame shift, as we've talked about before, would move the code. Remember, it's a triplet codon. So let's just say we had an A, a T, and a G, and let's go make it another A and G and the C. Let's just do that. So if we know that each one of these triplet codons codes for an amino acid once it's made into a primary transcript, but if we were to delete, okay, have a deletion of A, the new reading frame would be TGA, and then GC, whatever the next amino acid is, and that would change the reading frame, and now you're creating a whole bunch of new what? Yeah, new amino acids. If you've got a single base pair substitution, okay, if we bring back A, okay, ATG is the triplet codon. If I was to change it to, let's say, a G, now it's GTA, now we know that this triplet codon will be different, and changing the first two definitely changes the amino acid, but you do not change the next three or the following three. There's no frame shift. You would just change the code for just one amino acid, and that's why A is the best answer. A single base pair substitution would only make one amino acid become something else. Glutamic acid became valine, okay, in the gene. And of course, translocation of DNA from one chromosome to the other, that's kind of like a piece of DNA is kind of being moved around in the genome, but that's not what we're talking about. A piece of DNA is going to be longer than a triplet codon, and it's much more egregious in terms of how much is moved around. We don't have these pinpoint nucleotide substitution. You'd have whole parts of a gene possibly moved around. So A is the best answer. So let me speak about translocation one more second. Translocation, when you have sections of DNA code going from one chromosome to another. This sounds like crossing over like we would have in meiosis, but it's, as I said before, it's a little bit different or egregious in the fact that instead of just crossing over, when you're crossing over between homologous chromosomes, okay, you're not changing the position of where the gene is, as long as the crossing over is even, as they say. So most of the time, this crossing over of one part of a gene to another part of the gene, you're not changing the position of where the gene is. However, in translocation, you have basically crossing over between non-homologous chromosomes. So you have different chromosomes. So therefore, you're changing where that information is located. So we have two different chromosomes, and there is a translocation event where two different chromosomes are now changing the information on different chromosomes entirely, and now the gene's located not only in a different spot on the chromosome, but on a different chromosome altogether. Okay, and that would be a far greater scenario than changing one position, okay, as a singletide. If we're just changing one amino acid, translocation would be a much larger piece of a gene or multiple genes that are moving to a different location, to a different chromosome, and that would lead, possibly if you take a larger part of the gene away, a larger amount of, it have to be more than one amino acid changing. Okay, so in any case, 3 is A. Okay, number 4. In the year 2000, specimens of this animal, a green algae used in tropical aquariums, were found off the coast of California. They're native to the Indian Ocean. So this organism is known for its aggressive growth and ability to compete with seagrasses. It's currently on the international list of invasive species. Okay, which of the following best predicts the consequences of the introduction to this non-native species to the California coast? Okay, well, let's look at the choices. Without natural herbivores or competitors, this organism will grow rapidly and crowd out native species or producers. Boy, that seems to be the best answer going forward. And you say, well, why? Well, it's a non-native species that's aggressive. It's competing with other seagrasses, and basically it's competing for the same resources. And these seagrasses who have pressure on them, but maybe a keystone species or other organisms, now have more pressure on them by this what? Rapidly growing organism that may probably not have any competitors or natural predators that affect it. So what happens is that it will choke out the seagrasses because they have a new pressure on them that's taking up their resources in that niche. What does B say? This organism will have a hard time establishing because it will have to compete against native species which are better adapted to the environment. Well, they may be better adapted to the environment, but this new non-native species doesn't have pressure on it from other types of organisms in that ecosystem that keep it in check. That's important. So, all right, there's a keystone species in every ecosystem that keeps things in balance. And if this is non-native, that keystone species, or there's no check and balance for this organism. So it'll have an easy time, okay, competing for the same resources. This organism will grow rapidly, leading to an increase in diversity. No, there'll be a decrease, okay, in diversity, because it's going to eliminate and possibly kill a few different organisms that share the same resources. And because the current organisms that are living in that ecosystem are under check and balance by prey, predator, parasites, or their own little niche that's balanced is now unbalanced because they have now less of those resources. Okay, all right, so that doesn't make any sense for C. And then because it's not in the Indian Ocean, its natural environment, it will not be able to grow efficiently. That is incorrect. Okay, we've seen plenty times of non-native species grow elsewhere, and because of the non-checks on them, they can be detrimental to lower diversity. So 4 is A. Number 5. Okay, so number 5 is a classic prey-predator relationship graph where we have wolf, we have moose population size, and you can see clearly that there is limiting factors that level off these different populations at different times, but they're related to each other by something that we call density dependent factors. And of course, there are 4 that you have to know. 1 is disease, 2 is parasitism or parasites, which I think is related to each other. 3 is predation, which we see here. And then 4 is competition. Now, these wolves and moose basically are in different niches in the population, but what makes this a prey-predator classic graph is we see different times where the prey and predator are inversely related in their peaks and valleys. You know, if we look very carefully, quickly, we see that the moose are increasing, and so are generally the prey. And you notice that something affected the wolves here, and this could have been a disease. This could have been some kind of competition or a parasite, you know, or basically predation maybe by humans, but they declined, and because they declined, then we don't know what that is. The moose, because they are impacted by the predator of the wolves, increased until they reached a limit. All right, now why do they reach the limit? Probably because of the increase of the prey. or an increase of predation. But we don't know for sure, but definitely you see these guys cycling back. Now, as the predator, the wolves increased in size, okay, you can see the decline in the prey or the moose because the wolves are impacting that. So you see these up and down values, and you see classically in prey predators that one is up, one is usually down because they're impacting each other by one of the main density dependent factors, and that's predation. So this is screaming prey predator relationship. So we look at the choices, we look for that, and the wolves are predators of the moose, which were otherwise reproductively successful, and that is clearly the answer. Looking at these choices, the reproductive rate of the wolves is greater than the reproductive rate of the moose. Hard to tell without slopes. Both of them go up pretty steeply at different parts of the curve, so you really can't make that judgment. So that makes A a very poor choice. Also, mutualism is a word that just jumps at you. Ooh, they're giving me a vocab word, so maybe that's the answer. And mutualism, by definition, are based on when two organisms, not populations, basically benefit from a relationship where each one is helping the other. I don't know. The classic one is, I guess, if you look at rhinos, have birds that live on the backs of them and basically pull out the bugs, and the birds get the food, and the rhinos basically get the tick removal or bug or pest control taken from them. So that's a classic example, but that's not really what we're talking about here. We're talking about changes in population, not how organisms can mutually benefit from each other. So B, although they're introducing a vocab word to try to get you maybe, hey, that could be the answer, not the right answer. Speciation, not even close. These are already two different species that are part of an ecosystem based upon the prey-predator relationship. So D is the best choice, number 6. Okay, so 6 through 9 are a essentially cluster question, and we've got a graph here with error bars, and the error bars are plus or minus 2, standard error of the mean, which we should know real quickly is a reliability factor to show us that the value they're getting from an average is within this area. So they definitely came up with an average when they did a whatever test they're doing. They came up with an average value, and they are confident within 95 percentile that the actual true mean from that test is within this area. As you can see, big error bars suggest that the confidence is pretty low if the actual number can be anywhere within these two areas. Any case, one of the things you should do with these cluster questions is you should try to scan to see what they are and go to the questions as soon as possible. Many students are having trouble because they're spending way too much time trying to figure out what this is all about. So I'm looking at this, I see nitrogen influx, and if I can just scan this real quickly, we're talking about salmon and black bears. So salmon and black bears, and they're example of keystone species, which are really important species that may not be large in number but contribute largely to the ecosystem for many different reasons. So you have Pacific salmon and you have black bears, and you're saying that they're examples of often cited as examples. And any case, this is all about, if you look carefully, okay, of salmon migrating from their marine habitat to freshwater streams. They bring nitrogen and other marine derived nutrients. You need to understand that marine means salt water. So salmon come from, essentially they spawn or given birth from eggs in freshwater and then move to saltwater. But when they come back, they bring nitrogen back. And when they bring the nitrogen back, they call it nitrogen influx. So this investigation is a relationship between black bears, salmon, okay, and the influx of marine nitrogen. So let's just take it from there and go to the questions ASAP. If we have to dive back into these questions, we will. So 6, okay, states that which of the following statements is best supported by the data? Black bears are a keystone species. Salmon are a keystone species. Well, real quickly, they're both considered that in the reading, but the data doesn't show who could be a keystone species. Okay, so if you weren't sure, go back to the data and you will see that there's nothing here based upon. So we look at it carefully. We have neither salmon nor bears present, and this is a measure of how much nitrogen is going back into the fresh water. And so we have that. This is just the bears only. This is salmon only, and this is both salmon and bears. Okay, so none of this tells us if both could be keystones. Okay, so back to reality here. Up goes gravity, and C, black bear urine is a significant source of nitrogen. Okay, well, that would mean that black bears, okay, by themselves would bring a lot of the influx of nitrogen in the streams, okay, where the salmon are. So that would mean that the black bears, and again, if you're not sure, look at here, black bear only present right here. That's right here. And if you look carefully, the black bears are not significantly different in their nitrogen. So when you only had black bears only, there's no significant difference between black bears and having neither salmon or black bears. So there is no statistical significant difference. And why? Because the error bars overlap. So there's no difference between having neither salmon nor bears present or having the bears present. So that statement for letter C clearly suggests that this is not significant, it's insignificant. Okay, so I'm left with D, and I can just basically circle it and move on, but let's explain it. The presence of black bears and salmon correlates, which means that there is a significant difference in the increase of the nitrogen, okay, being influxed into that ecosystem there. And if you look, S and B is both what? Both salmon and bears present, and there it is right there. This number, although it does have error bars, the error bars are small enough, and if you notice, there is a gap between the bottom part of the error bar here. They don't overlap. So from this point, and I'm going to draw a big circle to represent nothing. This is basically our control. And this point right here, you can see that there is a statistical difference because the error bars don't overlap from this position to this position. So there's an increase of nitrogen influx. Considering the error bars, there is a significant increase. So therefore, I can say with confidence, that D is the answer. And again, real quickly, you didn't have to dive into all of those things. This is basically looking at the data points. And I know this part was in the reading, so again, you have to scan the reading, I suggest. Okay, so number 7. Number 7. Which of the following most likely describes how the interaction between the bears and the salmon influences nitrogen dynamics in the environment? Okay. Now, what was this study about? Okay, and again, you had to pick this up in your scan. You might have to reread it. Look carefully what it says. When salmon migrate from their marine habitat to freshwater streams, they bring nitrogen and other marine-derived nutrients. We read this once before. We first started looking at this, but it's important that we understand and highlight it. Okay, why? Well, because this nitrogen influx that we're measuring with this graph is the nitrogen that the salmon bring back. It's an influx. Influx means that something is going in. So the ecosystem by where the bears in the river go, nitrogen somehow, this element, very important in the development of any organism or the homeostasis of any element, because we have what proteins are made of nitrogen. So we have an influx in nitrogen entering and coming from where? From the marine derived nutrients. So it's basically that the salmon are bringing nitrogen into that ecosystem from the marine, the saltwater environment. So when you read your choices, and they say which of the following most likely describes how the interaction between bears and salmon influences nitrogen dynamics, okay, it's about how the salmon are bringing back. So when you look at choice B, when salmon swim upstream to spawn, bears migrate to rivers and deposit nitrogen from the forest. This is talking about how the bears are bringing the nitrogen that they have concentrated in their system from their ecosystem and bringing it to the same ecosystem. This is not about what? the salmon bringing it from the marine saltwater environment. Okay, remember the salmon spawn in freshwater, but then they go to the saltwater and basically develop there, and now they're bringing their larger bodies and nitrogen back to the freshwater environment. Bears are a reservoir for nitrogen, sure, but from their own ecosystem. It's not about from the marine influx of nitrogen. Bears urinate in rivers and streams where they provide nitrogen for salmon, but this is not the influx that the salmon were bringing to the table. So just by understanding that alone, we get rid of B, C, and D, and A, of course, is the answer. When bears consume salmon, they leave parts of the carcasses on the ground, which decompose, rot, and release the nitrogen into the environment by which they were collected by the salmon living in the saltwater environment. We know that decaying animals is a source of fertilizer, okay, for many types of growing animals, and people have buried, including my father, fish heads from fishing under the ground next to next to tomatoes for bigger tomatoes. Why? Because they decompose, and of course the fixed nitrogen, okay, the single bonded nitrogen, not the triple bonded, okay, is able to be utilized by the plants. And remember that nitrogen's important, of course, because it's part of amino acids that make proteins, and of course are the soldiers of our regulation of life. But they're also in part of photosynthesizers. Remember Rubisco, okay, the important protein that was in charge of fixing um, carbon from CO2 into carbohydrates, or the G3P that we talk about, which is the precursor for all other types of fats and oils and carbohydrates and storage material and cellulose that the plant needs for its own life. Okay, so 7 is A. Number 8. Number 8, which of the following pieces of additional data would help further investigate the relationship between bears, salmon, and the influx of nitrogen into the local environment from the what? From the marine environment. Okay, that's the whole point of this saltwater environment that the salmon have developed and grown, and have developed a lot of nitrogen in their bodies that they're bringing back to another ecosystem. Any case, so we're looking for an additional data to help further this experiment understand the relationship between the what? Between the bears, the salmon, and the influx of nitrogen. Okay, so if we look at some choices that are terrible, the experimenters should increase the number of bears in the area and measure the amount of nitrogen available for the uptake. This makes no sense because we're not talking about what? We're talking about nitrogen and bears, but we're leaving out how the salmon is part of this relationship. Remember, additional data. Keep rereading the question. It's about the relationship between the bears and the salmon and influx. This is talking about bears and nitrogen, but leaving out the salmon. See, the experimenters should set up a net to catch salmon. Great. And they're talking about nitrogen influx, but guess what? We're leaving out the bears. This is about the relationship of the bears and the salmon and the nitrogen. The experimenters should clear-cut trees from the area and determine how much nitrogen remains in the stream. So this is not even about bears or salmon. This is about the trees and how they affect possibly the nitrogen. So you can clearly get rid of B, C, and D, and A is your answer, because this one should remove the remains of the carcass immediately after the salmon are eaten by the bears and determine the nitrogen content. So we got salmon, bears, and nitrogen all in this answer, which is the only one that shows a relationship of the three. So 8 is A. You need to find a way to go fast on these easier ones. Okay, so 8 is A. Number 9. Number 9, if a dam is built downstream and prevents the salmon migration to the test sites, which of the following most accurately predicts the impact on the nitrogen influx? Well, again, let's not forget what this was about. Very quickly, we learned that when there was no bears or no salmon, this was the amount of nitrogen influx, and it was a big, big, big error bars going here. So low reliability, low confidence. When we had bears here, there was no statistical difference. When we just had the salmon, there was also no statistical difference. Notice these error bars overlap these error bars. So there's a great chance that both of these values could exactly be the same average. There's no statistical difference between the bears alone or the salmon alone. Only when we have the salmon and the bears interacting was there a statistical difference from this potential number anywhere in this area to this one because of the error bars. So there's only a statistical difference between the salmon and bears interacting in terms of the increase of nitrogen. So nitrogen influx will increase if you eliminate the salmon. No. How can they increase? It's about the influx that the salmon bring because of how the bears eat and maybe develop in their own urine or leave the carcasses behind in the rotting fish. So that doesn't make any sense. The nitrogen influx will decrease, yes, because there will be less bear-salmon interaction. This is what the entire problem is about. Okay, and the rest of them make no sense. Move on. The graph clearly shows this whole question is about the bears and the salmon are both contributing to a higher level of what? Influx of nitrogen than the control, which showed nothing. Remember, the bears by themselves and even the salmon by themselves, there was no statistical difference because of the error bars. Okay, so the only place where you had statistical difference was when you had the bears and the salmon interaction. And that's what this is about, choice 9. Number 10. Okay, so number 10, we've got the waggle dance question. So we've got some bees depicted in the diagram. Honeybees communicate the location of their flower patches, okay, to members of their hives with waggle dances that give information about the direction and distance of the flowers. Which of the following, okay, statements about how honeybees communicate the position of flower patches is most consistent with the model? So here's a model where they're giving you a lot of information. So you really can't look at this for 5 minutes and say, what is this saying? You got to go to the questions and try to see if the questions, at least I should go to the choices to see if the choices are going to give you some hints to make some sense of this. Clearly the bees are dancing in some sort of phase, and this is the time period, and there's an angle and there's flowers. Let's go right to the choices. Don't waste any time trying to figure this out. The number of repetitions of the waggle dance indicates the quality of the pollen source. Well, the pollen source, of course, is in the flowers. And from our diagram, I don't see how one has a better quality than the other since these pictures are the same. So there's no information to show the quality of the pollen source, okay, which would probably be how much pollen per area is given off. But even that, that being said, let's go to B, okay, and we see that the total area covered by any waggle dance leads to bees to target flowers. The total area. Now looking at the information above, there's no information about the area in which they dance. This is a waggle phase, which looks like it's a time period, okay, in their dance. So their dance has different time periods. This one's shorter, but I don't see the area that they cover in their dance. Like when I dance, I cover a large area because, well, I'm just a bigger person, but that's a different question for a different day. So I get rid of A and B because that information is not given to me in the diagram. The angle of the waggle phase relative to the vertical plane indicates the position. So you're gonna have to look. The angle, all right, to the vertical, well, one's going to the left, one's going to the left. I'm not sure if I quite understand it. That could be an answer. Let's see if I can eliminate D and see if that might be the answer. The farther the target flowers are from the hive, the longer the waggle phase. So again, C could be right. I couldn't really interpret this really fast. I'm not sure what this means. Waggle phase relative to the vertical plane indicates that's a lot of information, and that could have been right. We're not sure, so I just went to D as a test-taking strategy to see if that might be something that's easier for me to figure out, and this one is. And what you see is that when flowers are closer to the hive, this is 160 meters, the waggle phase, the time of the phase of the dance, is shorter. And when the distance between the hive and the pollen source is longer, 500, so is the dance. The dance is longer, 375 milliseconds. Those are fast dances, and 150. So the time is relative to the distance. That's what this information is telling me. And there's no way that I could have taught you any of this other than just looking to see if in fact the information in the graph is representative of the choices, and D is the best choice. So the length of time of the dance is definitely relative to the distance to the pollen source. So 10 is D. Number 11. Okay, so number 11. Eye pigment in a particular strain of fly is determined by two genes. An autosomal gene, this is a gene from the non-sex chromosomes that controls the color of the pigments in the eye, has two alleles. Has a dominant, okay, R that results in red eyes, and a recessive that results in sepia eyes. Okay, so this right here is information of a non-sex linked trait, okay, or allele. These are on the regular chromosomes, okay? And then, of course, they're going to give you some information here, a sex-linked gene that controls the expression. Now, what does this mean, the expression? You need to understand, to control the expression, that means that this gene will affect whether or not we get the red or sepia colors at all. And it has two alleles. T that allows for the expression, meaning you can have the red or the sepia. I'm not sure what the sepia is. And then you have the recessive or the broken gene that does not allow for the expression of the colored pigments. Okay, if it doesn't allow, you're going to get white eyes. White eyes is the eyes you get when you don't have a color, which means you must have the recessive allele in your sex-linked gene for the expression. So the expression is sex-linked. The actual color, okay, is non-sex linked. So here comes the question, since we're going to combine these two ideas, which of the following represents a cross between a white-eyed female and a red-eyed male? So we have a female, okay, and females, of course, are double X, and we have those in our choices, and the male is XY, so that doesn't come into play. But we know that the female is wide-eyed, and as our reading here, the wide eye lacks the expression. So that means we're going to have what? The recessive allele on the sex-linked trait. Okay, since it's a recessive allele for the mother, you're going to need to have both of them there. So when I look for XX, oh, there's a white-eyed mom, and so there is another white-eyed. These are both little R's, I think. Let me just look carefully again. No, this is a T, sorry. And this is an R. And so what they're doing in A and B is they're trying to see if you knew the difference between the sex-linked and the autosomal. Notice the T little t here. They said right here, party people, and you need to focus here, that this was the sex-linked part. That means that these alleles, T and little t, have to be on the sex chromosomes. So we get rid of B and A collectively because they're putting the T and little t on a regular chromosome, and on the X, remember this is the X chromosome, they're putting the R's, and the R and the big R, the little recessive R for sepia if you have a homozygous recessive, and the dominant for red eyes has to be on not on the sex-linked gene. So we get rid of A and B. Let's look at C. Well, we have big T, if you can see here from these two X's. That means that we're going to express whatever the color are. So double R is homozygous dominant. This is the red on the autosomal non-sex linked. This will be red, and this sex link expression control is going to be what? It's going to be big T, which means you express the red. So C, this is going to be a red, or double R is going to be red-eyed female. Okay, so forget C, so D has to be the answer, but let's understand D because I can see this being a part 2 question where they actually want you to write it in. So first of all, we look at the female, and if you look carefully, you see X, little t. And what does that mean? Homozygous recessive, and that means that's the broken gene. You're not going to express the red color, and that's going to give you, I'm going to write this in white, watch it, white eyes. You see that? No, okay, not funny. Okay, so this is the white eyes because it's not going to be expressed. The expression of the color in a non-sex-linked chromosome, the autosomal, doesn't get expressed because the sex-linked expression gene is broken. It's recessive. So that's going to be your white-eyed female. And then your male, if you just want to have fun, red-eyed, it's red because of big R, little r, the dominant gene in the autosomal part expresses the red. And carefully understand that the male only has one X, and that X is T, and it's big T. The Y doesn't carry that allele or that gene, so that means you're going to express, the big T means you have the expression gene on or working, and therefore you get the red-eyed male. Okay, so number 11 is D. I love this little question. Number 12. So number 12, I find this question number 12 to be more of a question that is something that you had to know from the course than any other question I've seen in the multiple choice so far. Meaning, I really think you had to know something about transduction. And we've seen this in other parts too. So boy, that's terrible. Transduction, okay, is the concept that this question is about. But let's go read the question. Some strains of bacterium, Streptococcus pyogenes, secrete poisonous substances called exotoxins. The genes encoding exotoxins are thought to have originated in bacteriophages. We should know what they are. These are viruses that infect bacteria. So somehow strains of bacteria, okay, are able to make these exotoxins because somehow viruses that infect other bacteria, okay, have somehow led that genetic code into certain bacteria. Well, that idea is what's called transduction, meaning a virus with again, that protein coat hits a receptor and basically injects its DNA. Now that DNA it can inject to take over the cell to start the lytic cycle, may or may not create a lytic cycle. And you may say, well, why? Well, because bacteria have these endonucleases, these little proteins that can actually cleave up the foreign DNA that's injected. That's where we get those types of, that's probably the first type of immune system that we know of, these endonucleases, which we call our restriction enzymes that we use to cut up the fragments of DNA into smaller fragments, and then ran them under ran a gel or undergone gel electrophoresis to see the differences in how the bands move based on size. But in any case, the idea here is that DNA was cut up, and so you got these little fragments of DNA, and some of these guys can get incorporated to the plasmids, okay, of a bacteria, okay. And if this piece of DNA gets incorporated to the plasma, and it came from another DNA that was that this virus was made from in another lytic cycle, and if that piece of code is a code for the exotoxins, that's one of the ways how there's horizontal gene transfer, okay, or horizontal DNA transfer from one bacteria to another. One of the ways is through these bacteriophages with these viruses that try to infect, lose because probably there's a restriction enzyme that cuts them up, but some of these pieces, okay, are able to still find their way into and join that plasmid, that the bacteria's DNA that it still has. So transduction is again an example of horizontal gene transfer, and that's what this question's about. And if you look at the choices, bacteriophage DNA become integrated into the bacterial chromosome. So that's basically what this entire question's about, is about transduction. Okay, one of the ways that bacteria can deliver, at least can have basically other DNAs other bacteria's DNA incorporated in it and increasing the genetic diversity. Okay, of course, there's other ways as well, conjugation, where you have direct DNA transfer from two different bacteria, of course. Um, and also transformation, where DNA could find its way into through the cellular membrane. Now, we did transformation with our P-glow protein with the genetically engineered plasmid, and we basically heat shocked the cellular membrane of the bacteria, the E. coli high school bacteria that we're using, to get this incorporated into the plasmids, or create a basically create a new plasmid chromosome in the bacteria. So transduction, conjugation, and transformation are the three ways you can have horizontal gene transfer from one bacteria to another. And that's important that you know that it's popping up. And 12 was all about transduction. Number 13. Okay, so 13 to 16, another cluster question. A lot of reading here. You don't have the time. If you want to scan what's going on here, they're talking about hybrids, similar sex chromosomes. Oh my gosh, there's all kinds of different insignia. A lot of info here, but just quickly say, over many generations, two populations of the same species can diverge into separate species. Okay, so we're talking about divergent evolution, where basically because of reproductive isolation due to probably geography, okay, they go into separate species. Okay, and there's four different stages of speciation. I'm just basically scanning this to have some basics. Okay, and I look at what they're showing me: fertility, population, and this is the mating between individuals in initial populations, and this is fertility and the matings between diverging populations at an intermediate means in between stage, and then we have at late stage speciation, which means we have reproductive isolation, and we can see the fertility has changed there. So I'm just kind of scanning it and going quickly. I may have to dive into it deeper, but let's go to the questions. Based on the model of speciation presented, which is about isolating two populations and over time having reproductive isolation, which of the following describes the most likely consequence of the populations over time? Okay, so hybrid individuals are less likely to pass their genetic information on to subsequent generations. Boy, this is something you see in hybridization all the time. I'm sorry, you see in speciation, that there is low reproductive success between the two populations, to the point where there's no longer any success. So this is jumping out as a possible answer. It is the answer, and you should know that in speciation, the hybrids are not viable, which means they cannot have offspring very well. And that's, even though you may have crossbreeding between two different populations or two different species, or eventually, okay, it's important to know that speciation is going to occur when there is reproductive isolation. So if you look quickly through the graphs, and I get it, there's so much information here. This is, to me, just eye overload. But if I can focus in on fertility, in the early mating between two populations, you have fertility, which means fertility means that the hybrid were able to reproduce themselves. And this is in the initial populations. Okay, now when there was crossbreeding, after there was more geographic isolation, possibly, there was what? Less fertility, I should say. So there was still some, but you can see over time that there's a lowered amount. And then we get to the late stages of speciation, when these guys are now almost completely diverged away or have diverged, there's absolutely no fertility. So the model they're presenting is that the reproductive isolation is that the offspring cannot reproduce, and therefore these two populations are now two different species. And that's what A is speaking to. B, hybrid individuals living together are physically unable to mate. Over time, living together are unable. There's no information about when they live or not live together. So this is it's a choice that you say, ooh, they can't mate to each other, and that seems to be a good second place finish to A, but not a good choice there. Geographically isolated populations gradually acquire similar heritable traits? No, they go in the opposite direction. They have different traits over time. Okay, and therefore that's what eventually those traits lead into a reproductive isolation. So that's going the opposite way of how you should be thinking. Differences between hybrid individuals of a species disappear over time. More differences increase over time as two populations become two species and are isolated. So this is also going the opposite way. So A is clearly the right answer. And you probably, if you understand speciation and hybridization between them and reproductive fitness that cannot exist between two species, I believe you could have answered A without even going into the graphs. All right, again, it takes practice. Number 14. Okay, number 14, which of the following best describes the reason for excluding? Including would mean you use it. Excluding means you would not use something when calculating the allele frequencies of two interbreeding populations. Now, why would we want to calculate the allele frequencies? Well, we want to see what the percentages of certain alleles between the two populations to get a better feel for if speciation between the populations is happening. If the allele frequencies, the amount of different types of the same type of expression of the same gene, changes between populations, there's evolution between them. There's divergence between the two populations. So that's why they want to do that. But they want to, why would you not include hybrid males? And they're saying go to figure 2. So let's go to figure 2. When we go to figure 2, we see that the males, and if again, best way to see this is the males are what? These are the interpopulation male hybrids. So we see in figure 2 that we have the male hybrids, and notice their fertility. They don't have any fertility. Therefore, they can't contribute to the frequencies of the next population. Okay, so we want to look at the allele frequencies here and see how they differ. And so we can't use these guys because they're not going to pass on their genetic information. So using their alleles would be basically cluttering up our data because we're trying to figure out what alleles are possible. So they're not having any genetic information can be passed around because they're infertile. And that's all that's about. So if you look at your answer, which of the following best describes the reason for excluding the hybrid males? Well, because they're what? They're infertile. So sterile individuals make no genetic contribution to the next generation. All right, and that's why C jumps out. There's no real reason to go over the other choices. Okay, and that's what that was about. All right, so number 15. Number 15, in a separate investigation, individual mice from two populations in nature are geographically isolated from each other and mated in the laboratory. The hybrid offspring were then mated with individuals from either of the original populations. Only the female hybrid offspring were fertile. The experimental results are most consistent with what stages that are depicted in the model. Boy, we were just doing that with 14. Which stage were only the female hybrid offspring fertile? Which means the males were infertile. And if you look, that would be figure 2, and that's the intermediate stage of speciation. We were just there explaining the last problem where all the males, okay, were infertile, where the females were still fertile. So clearly, okay, all the females, and again, if you don't see that, you go to your legend, and your females are here, and here's your female interpopulation, and all the females are fertile, whereas the males are not. Okay, so those results are from the intermediate stage of speciation. This question is just reading the graph, interpreting the results. Pretty simple, and that's why B is the answer. 15 is B. Number 16. Number 16, using the model of speciation and applying it to a different population, which of the following outcomes is most consistent for different species in which the males are homogametic and the females are heterogametic? Now, if you don't know what these terms are, and I would not have known them either, okay, unless they were in the reading. So we're going to go back and dive into this to understand this question. Before we do so, though, understand usually these types of questions are very easy in the beginning and reading the graph, and then usually the last couple of questions in a cluster question like this is, okay, 13 to 16 was the cluster based on the information in the graph. These later questions are really a little more tougher. So if you don't have time to dive in, leave 16 blank and go back to it. Go after the easy ones first. Any case, let's go back to it to show you where they talked about this. And again, it takes time because you have to dive back into it. But they said earlier on, or you have to find the males represents the model are heterogametic. And now we know hetero means different, okay, which means they have two different sex-determined traits, thus X and Y. And then there's homogametic, which is what mostly females are like, where they're double X. So what they're saying is, in this problem, okay, using the model of speciation, which is basically reproductive isolation by the lack of fitness of the hybrids, which of the following outcomes is most consistent for a different species in which the males are homo? Somehow the males now have the same chromosome, and where the females have two different. Now you may say, well, why would that make any difference? Well, if you think about it, if there's reproductive isolation between species, there's something about the sex genes. And since we know that the X chromosome is so much bigger than the Y, remember the Y basically only has the male characteristics, and certainly the X chromosome being a heck of a lot bigger, okay, has the female characteristics and other genes, okay. The reproductive isolation problems are more rampant with the female's end. Okay, so now you may say, well, how do you know? What if the sperm made by spermogenesis is not viable and can't get the egg? Certainly there's going to be times where the Y chromosome or the genes on the male end of the reproductive part, okay, could have a mutation or some kind of change where the sperm can't fertilize the egg. Certainly that can happen. But remember, this is a smaller chromosome. There's less information. So the chance of the problem occurring because there's more code here is on the X. If you still don't like that idea, okay, if you look back on the data given to us, where was there a stark difference in a type of viability or fertility? In the late stages, none of the hybrids were fertile. But if you go back to the intermediate stage, which the couple of questions looked upon, notice all the females were fertile. So it seemed to me that the issues were with the males. Now, why with the males? Well, if we assume that the problem is on the X because it's a bigger chromosome and there's more chance of mutation occurring there. So if the problem is on the X, remember, to be a male, you only have one X. So if you only have one X gene, and that's the recessive or broken gene, you don't have another X to have a dominant. Whereas a female, you could be a carrier by having a problem, but you can have the dominant good gene there to mask and to overtake the broken gene. So that's what this is speaking to. And if you didn't see that, that's okay. All right, it's important you get that. So the males, so this is kind of like a sex-linked problem with reproductive isolation. And so that's where this is coming from. So if the males, okay, had the same, I would say, set of chromosomes as the females, they would actually have the genetic advantage, because if the males were homogametic for whatever reason, they would have two chromosomes that were the same. And so if they had a broken chromosome with a broken gene, they would still have the dominant one that would mask it or overtake it and still have the number of genes being transcribed from this good chromosome that's to still maintain reproductive viability. Whereas the females, if they have two different chromosomes, okay, all they would need is have one broken gene or recessive allele to express it. Okay, so if you were to change the scenario based on what we said, sterility would appear in females before appearing in males, because the females now would only have one chromosome um, that would, of course, have an X, and and therefore they wouldn't have another chromosome that was similar to be dominant to again mask or take away the recessive part. So that's why 16 is A. I'm not even getting through any of these, don't make any sense. If you look at letter D for a second, the population would reach Hardy-Weinberg equilibrium. Hardy-Weinberg equilibrium, party people, is nothing more than a set of conditions where we do not what? We do not have evolution, like no mutation, no gene flow. Populations are large, okay? Those set of conditions, no mutations, okay? All right, and random mating, of course. So none of these make any sense. Behavioral isolation, okay, just means different behaviors create the what? The isolation between the two different species. But again, A is the best choice by far when you consider about two different chromosomes, okay, and the sex-linked characteristics that create the reproductive isolation. So 16 is A. Number 17. Number 17, a scientist is evaluating a proposal for raising large numbers of fish in ocean pens for human consumption. As part of the evaluation, the scientist is designing a plan for investigating how the fish in the ocean pens might affect nearby ecosystems. Okay, which of the following is the most appropriate factor to use as the dependent variable? Right, the dependent variable is the one that we put on the y-axis to measure to see how much it changes, okay, uh, due to um, how the dependent variable interacts with the independent variable, which is what we put here. Okay, so we look at D, the amount of time it takes for the fish to digest a meal. So you could have a time, okay, and different meals down here, and you could measure the different times based on different meals, and that time would definitely be a dependent variable, but that is not the dependent variable that we would need for this type of investigation to see how fish caught up in pens in the ocean would affect other ecosystems. So this would be a dependent variable, but for this doesn't make any sense for the study that we have. The types of fish that are preferentially consumed by humans, I believe that's more of a independent variable. See how the different types of fish, they would go here, and then we could see how they're affected somehow by some kind of dependent variable. So I see that the types of fish would be an independent variable, okay? And B, the water temperature in the habitat. Well, the water can't be a dependent variable because the water temperature is not going to change in the ocean based upon the types of fish I have. Okay, so the water temperature is an independent variable. Okay, so that doesn't make any sense. So we're left with A, the amount of metabolic wastes in the water where the fish are being raised. What are these metabolic wastes? This is the nitrogen influx in that area. So the nitrogen or the urine they give off, or whatever waste their excrement that gives off, okay, we can say that how much this could be the amount of the wastes that we're going to depends upon how many fish. So we could have different numbers of fish, okay, and we can measure what happens to the metabolic rate as the number of the population increases. So again, we're measuring on the y-axis, and certainly A is a good independent variable. I'm sorry, dependent variable that depends upon the type of fish possibly and the amount of fish and how much waste nitrogen is being given off. And of course, too much nitrogen is not a good thing because it could spawn, for instance, an algae bloom, especially if you've got waters like our sound or our bays that have some algae in them. So increasing the nitrogen can act as a fertilizer for them to grow exponentially larger than they should, and then that could encroach on other niches and cause problems, as we've seen in our own waterways. 17, best answer is A. Number 18. Okay, number 18. Epinephrine is a protein hormone found in many animals. Epinephrine stimulates a signaling pathway that results in the breakdown of glycogen in liver cells. Which of the following describes the initial steps of the process where epinephrine stimulates glycogen breakdown? Well, this is going to be, for the millionth and one time, the reception, which is basically a chemical signal hitting a protein receptor. Then you're going to have transduction. And transduction is that cell signal pathway where we have a cascade of chemical reactions and then a response. This is what this whole thing is about. So A, epinephrine binds to a cell surface receptor, loving that. The activated receptor stimulates production of the secondary messenger, okay, which goes on. So that screams to me something that should be something we should know and move on. We did a report on epinephrine. How about that? Epinephrine binds to a cell surface receptor again, so this is a good possibility also. The activated receptor catalyzes the conversion of glycogen to glucose. No, it doesn't do it directly. The receptor doesn't. The activated receptor catalyzes the conversion. That would mean that the receptor on the outside of the cell, okay, all that occurred on the outside of the cell, it has to go inside the liver cell. Besides that, you're not talking about the transduction, the cell signaling pathway that must occur. CCAMP, of course, is part of that cell signaling transduction pathway. So that's why A is a better answer. C, epinephrine diffuses through the plasma membrane. No, it's too big of a compound. Only very small nonpolar hormones like testosterone. You would never have to, C would never be the answer because it'd be something you'd have to know. Okay, and then D, epinephrine is taken into the cell by endocytosis. Endocytosis is kind of like how the amoeba engulfs its food by making a basically pushing its cytoplasm over and to engulf it to make a vacuole. That's basically, and that's not how our cells work. We have a phospholipid bilayer and definitely different channels, but we have, this is a cell signaling pathway where we have receptors that cause a cascade of signals based to a response. So this is not how that occurs. This would be more like how simple organisms like amoeba engulf, okay, their food. So this would be choice A, number 18. So number 19 is an interpretive model type of a question where you have to look at the picture and make sure you kind of understand what it's trying to say. We have this phospholipid bilipid layer here, these phosphate groups and these hydrophobic nonpolar tails. We have these channel proteins. We have some small extracellular fluids that are able, these small molecules that are able to get through into the intracellular inside the cell by going right through. They're probably either very small and non-polar. And then we have these channel proteins, these integral proteins that span the entire width of the cellular membrane, where these molecules can fit in these certain channels that are specific for it. And both of these are moving by their concentration gradients from high to low. So these are going to be passive transport, no ATP is required. And here we have movement from low concentration to high concentration. This would require ATP. And this also is another type of channel protein that requires ATP to make happen, and this is of course active transport. In any case, let's look at the choices to see which ones make any sense. 19, which of the following scientific questions is most relevant to the model represented in the figure above? Is ATP required for the transportation of sugars across the outer mitochondrial membrane? Well, first of all, how do I know which ones are sugars? They'd probably be bigger than the smaller ones. But a mitochondrial membrane, okay, so how do we know it's a mitochondrial membrane? All right, so these are very specific questions that are too specific for this type of very, very vague and general diagram. So too much specificity for this type of question. B, do the types of phospholipids? If I look at my diagram, the phospholipids are all the same. These phosphate groups are here. There's no difference in them. So these are different channel proteins, but certainly, uh, this could be a pump of some sort, like a sodium potassium pump. But the point is, there's no differences in the phospholipids. They're all the same in my diagram. Which molecular substance is actively transported across the plasma membrane? Okay, so we can definitely answer that question by looking which one is going against the concentration gradient. These three are going uh, um, against the concentration gradient because things normally flow from high concentration to low. Just by hook or crook, having more on one side will find its way into the channel than the reverse. So this is going against the gradient because these guys are going to want to find the way back through the opening because there's more of them, so they collide more often. So it's going to take ATP to go against that gradient. And so that's a better question. Okay, the active transport. So C is the answer. In temperature, there's nothing in our diagram above that even speaks to the flow of materials in and out of a cell, in this case into the cell, by changes in temperature. So C is the best answer by far. Number 20. So number 20, we have an experiment between how well um, pea seeds and melon seeds will grow in um, compost. And so they gathered some data. They had 20 pea seeds and 20 melon seeds. Notice right away that that's a low N number. So the N is equal to 20 for both of them. So it's not a big sample size. And of course, they recorded all this information. What they found, the results were that in all treatments, the pea seeds germinated more than the melon seeds. Okay, also, there was a week of rainy weather, so it stayed wet and moist, which is if it was warm, wet, and moist, it's a good conditions for fungus. And there was a fungal growth at 4 weeks. The melon plants were definitely, there was an observation that we saw from our eyes that there was a fungal growth. Now, when that fungus started growing, probably in the week of their rainy period. Okay, so in any case, melon plants grew longer than the pea plants, but many melon leaves showed signs of yellowing as the weeks passed because they were inflicted with some kind of fungus disease probably. Okay, that being all said, the students concluded that the pea plants grew better in compost than did the melon plants because their data showed that even though there was a fungus growth on the melon plants and there was some infliction of some sort of disease, more pea seeds germinated than the melon seeds. Okay, now, which of the following best addresses the validity? Now, the validity would mean the idea that we can believe the outcomes are strong and that there's a high confidence in our statements. So which best addresses the validity of the conclusion made by the students? The students' conclusion was pea plants are better able to grow better in compost than melon plants. Look at our choices. It's one of those questions where you're going to have to read the choices to see which is better. The conclusion is valid because the independent and dependent variables were controlled. Now, I have an issue with this because of the infliction of the disease or the fungus growth, however you want to think about it. We did not control for that factor. Maybe that fungus disease was contributing to the melon plants not germinating as well. Okay, there was another factor here. So things were not controlled. There was another variable at play here. So I'm getting rid of this choice. The conclusion is valid because the experimental design included a large sample size. We just talked about the N equals 20. That's small. Okay, so that size is too small. When it comes to seeds, that's really small when you think about it. The conclusion is invalid because other variables in the experiment, both biotic and abiotic, affected their results. I'm loving this because there was another variable in all of this, and that was the fungus disease. Now, what's the biotic? The biotic would be the fungus. Okay, the abiotic, okay, would be that it's damp. Maybe the damp weather caused the melon seeds to rot. If you ever planted seeds in wet weather, sometimes they rot. So some seeds are able to have a thicker coat on the outside of the seed. Remember, the seed, of course, has the embryo that's been fertilized, and of course some stored um, nutrients, usually in the form of carbohydrates. When you pop popcorn, you're actually exploding that carbohydrate. But the harder the cover of the shield, or the harder the seed coat, the easier it is for the seed to combat the what? The moisture. If there's too much moisture too fast to dissolve that seed coat, okay, sometimes that may damage and cause the seed to not germinate as well. So we have damp factors that we're not controlled for, and the fungus. All right. So the fact that just the compost, remember this was just about the compost and how it affects the what? The two different types of seeds, yet we had a disease that may afflict one type of organism better than other. And then of course one organism might be better suited based upon its evolution for damp weather when one isn't. So these two other factors, biotic and abiotic, contributed to having lack of control of the other variables. Remember, when we were trying to have a study, we're trying to keep the other environmental conditions constant for both. And in this case, we had them in play and they could have affected the results. So C is the best answer. All right, and D makes no sense because it's not about the units of measurements. So number 20 is C. Number 21. Number 21, butterflies of the genus Coleus live in the Rocky Mountains where they experience a wide range of temperatures. Okay, so we're in the Rocky Mountains, and of course, in mountains, you could be in a valley, you could be on top. There's all different types. You can be on the sun side of a mountain. So there's a wide range of temperatures or ecosystems within this area. Different variants of a particular glycolytic enzyme. So we're talking about glycolysis, or an enzyme that is helpful in glycolysis. And of course, glycolysis, okay, is nothing more than a what? An anaerobic means of gaining energy through the breakdown or the catabolic pathways that breaking down, let's say, glucose, as we talked about, into energy. It's an energy coupling type of metabolic reactions. So anaerobic pathways without oxygen. This is the fast and older system. Now, we know that there's 10 steps to glycolysis, but we're talking about a particular enzyme in the flight muscles that are optimally active. So it's different variants of a particular glycolytic enzyme. So there's probably one enzyme responsible, as we talked about, that's an on pathway, that talking about an on-off switch in glycolysis. There's a particular step that we've talked about, and you don't have to know that, but there's a particular enzyme that once it is active, the whole pathway can go. But they're saying that this enzyme is active, optimally active at different temperatures, which makes sense. In the mountain, there's different places in the mountain where they can be warmer or much colder, and these are extreme, actually, temperatures. 29 and 40 degrees Celsius are extreme different temperatures. Any case, so we look carefully within the same population. Some individual butterflies fly most effectively at 29 degrees Celsius, while others most effectively fly at 40 degrees Celsius, which is really way over 100 degrees Fahrenheit. Still, others can be equally active at both temperatures. Okay, so we definitely have variants of the gene that definitely make two different forms of the gene, of the protein, for this enzyme. That's an on-off switch for the entire glycolytic pathway for the butterflies to fly. And definitely having two different temperatures selected for, probably because they're two different positions or two different places where these butterflies, okay, basically live. So it seems to me there was some type of isolation between these populations at some point, okay, where they had selective pressure based upon temperature to single out in a particular allele that was in low frequency. So natural selection was at work here with this butterfly, okay, either through a little bit of separation from a species, or these two species coming together recently, okay, and making a new species. Either way, clearly there are two forms of the same enzyme. And of course, there's still others, um, um, butterflies that can be equally active at both temperatures, to showing that there's multiple alleles for the same gene to make this particular enzyme. Now, which of the following claims is most consistent with the observed butterfly behavior based on this understanding? Okay, well, looking at my choices, I can see that butterflies that express two variants of the enzyme are active over a greater range. This jumps at me as well. That sounds really good. They express two variants of the enzyme, okay, that are active over the greater range. Maybe it's not three alleles as I'm thinking. Maybe it's basically I've got an allele that's optimally very good at 29, and then this would be number of individuals, number of butterflies, okay, and this would be the activity, or yeah, I gotta say activity, I guess, the number of flights of butterflies over a particular time period. So this would be for the 29-degree group. As you can see, I'm just drawing a bell curve. I'm making assumptions. And then also we're going to have some that are more active over the 40-degree temperature change. And so if you think with me, even though I initially said maybe there are three alleles, now that I think about this, you can see that you're going to have so many alleles in this camp at 29 degrees Celsius, and then of course you're going to have many alleles, okay, you're going to have many butterflies that have this variant, and so you can see you're going to have optimal activity at these two different temperatures, but you're also going to have a range of individuals who can do both because of the overlapping. So I like A, it jumps at me, and I can see why you'd be a little unsure about that. So you go through the rest of the choices to strengthen your position that A is the answer. That's what we do here. We're going to use the answers basically to see which one's the best. Because again, they're saying which of the following is most consistent. So butterflies that are active over a wide range of temperatures produce greater amounts of enzyme. This is a horrible but an effective distractor, because you say, oh, great amounts of enzyme, therefore I fly better. But remember, the selective pressure was the temperature. Okay, by saying it's about producing the greater amounts of enzyme, then the amount of temperature would not be a factor. They're not talking about temperature as a factor in this choice. Okay, you're just saying the being able to make more of the enzyme makes you have greater glycolytic or anaerobic type of means of gaining energy. And certainly that's an advantage. But again, the reason why we have two groups of butterflies that are optimally active glycolytically at two different temperatures had to be due because of selective pressures of where they originated. Okay, so again, this is not speaking to temperature as a factor. Temperature has little effect. That's just going the opposite of our thinking here. Temperature was at some point a selective pressure, as I've said. And D, butterflies that are active in warmer temperatures produce more offspring. Okay, you may say, well, this has nothing to do with it. True, it's not the correct answer, because active in warmer temperatures means that we would only have, if only the ones that were able to produce more offspring, which is which basically is how natural selection works. There's more fitness, okay, for the population, or the individuals of a population who have the gene that makes the enzyme that's more active at 40. And if that was the case, you wouldn't see any of the 29-degree range of activity. So clearly when I think about it, there are just two different genes, or two different proteins being made because two variants of the same gene that are built in a certain way. One can withstand higher temperatures before denaturing. Ooh, what's that word? Denaturing means at higher temperatures, as we've seen with the catalase lab, higher temperatures make, okay, molecules vibrate more. And remember, amino acids make these tertiary structures, okay, by sticking together to other amino acids by their R groups. So you have an R group and you have another amino acid chain, and they fold together by the R groups. I should draw an R group here and get rid of that. And they interact and they attract. So higher temperatures, higher energy can basically pull these attractions or break these attractions and basically take the shape of the protein, which is in some kind of folded shape that occurs finally in the Golgi and endoplasmic reticulum folding occurs, but with higher temperature, breaking these attractive forces from the R groups would change the shape. Changing the shape of the protein changes the shape of the active site and therefore will render it useless at some point. In any case, the differences in these temperatures are due to, okay, there are different ways that the two butterflies convergently made two different enzymes to be more suited for their environments. So to say that one would produce more offspring would mean you would only have eventually at some point just one variant, and you wouldn't have the greater number of individuals in the current population that have an enzyme that can work at this lower temperature, and thus you wouldn't have both of them working. So A is clearly the best choice. All right. Again, the way that I'm explaining this, you don't have all this time to be thinking that much about it, but this was talking about two variants, two different temperatures. Okay. And the ones in the middle, okay, are the temperature range in the middle is due to having both of them, okay, having that range. Number 22. Number 22. So number 22 is a Stanley Miller question, and that was the experiment where we took known atmospheric gases at the time and of the early Earth. It's sort of a primordial soup hypothesis, meaning that if we have um, the components, the raw components in our atmosphere, and given the conditions of the early Earth, which is plenty of heat and plenty of, let's say, energy in terms of spark due to maybe electrical storms, that energy of the early Earth with the again, these are hypothesized types of molecules that were available at the time, that they would make precursors, and that's important, precursors, okay, to life. And now these precursors would be organic, and the word organic means that they're usually some sort of carbon-based precursors. And what's carbon-based precursors to life? These include nucleotides, right? And nucleotides, of course, result in RNA, which is, remember the RNA hypothesis, which is the early hypothesis of how hereditary information was passed down from the first living organisms. Eventually DNA. Of course, other types of carbon-based organic living types of molecules are amino acids. And amino acids, of course, linked together make the proteins, and the proteins, of course, or the soldiers and the doers of the metabolic or life-sustaining reactions are all run by these proteins. So carbon-based, of course, are fuels, or the energy that we get from life are carbon-based, the glucose and so forth, lipids and carbohydrates. So the carbon-based type of compounds that are precursors. So when you took the earlier compounds that were available with some energy and gave them time, the Stanley Miller experiment showed that you would make amino acids, you'd make some nucleotides, and you'd make some other organic precursors that are carbon-based, and that we see today in life. It did not spurn life. Life did not come from this experiment, just molecules that we find in life. And of course, in order to understand how life starts, we definitely have to understand that we have to have these molecules available. And we can see that they can be made just by random events using heat and the the basic gases that we think that were available at the time. So in any case, after this experiment using two different groups of gases or a different arrangement of gases, okay, with the same setup, this isn't really important that you understand all of this connectivity. It's about the gases with energy over time creating these precursors. That's what the entire experiment's about. Let's look at the choices. Which of the following statement best justifies the claim that the conditions in at least one of the experiments could generate molecular building blocks? essential for life. I guess it's a better way to say precursors. Okay, well, let's go to the choices again. It's all about the choices here. All right, carbon dioxide gas and water vapor in experiment one could spontaneously produce phospholipids required for the first life forms. Now, if we look at figure one, we have nitrogen, carbon, we have ammonia gas and water and CO2. To make a phospholipid bilayer, we notice we have the phosphate head, and then we have this tail made of carbons and hydrogens. So the phosphate head, okay, is this phosphate molecule, all right, and we know that it's polar. It's got charges, and these charges are attracted to water. That's the hydrophilic end of the phospholipid bilayer. But you need phosphorus. There's no phosphorus or P in any of these early gases, and you just don't create an element out of thin air. It has to come from somewhere. Okay, so A we get rid of because in order to make a phospholipid, which is the membranes, remember that important membrane that keeps the life-sustaining reactions separated from the extracellular, from the intracellular, okay, we need to have phosphorus, and there's no phosphorus in that first flask, so we get rid of that. B, the nitrogen gas and ammonia gas in experiment one could provide elemental nitrogen required for the formation of amino acids. Now, they didn't say you would make amino acids directly from just nitrogen and NH3, because amino acids, as we've seen before, or is this Cu group at the end, you know, another carbon with an R, and then you have a nitrogen. Okay, and this is the basic formula for an amino acid. Okay, and so the different R groups basically change the type of amino acids, but you have carbons involved. But not saying to make an amino acid you have to have nitrogen, or to make a complete amino acids you need carbon and hydrogen and nitrogen. What they're saying is, in order to make an amino acid, which must have nitrogen, okay, that could have come from or provided by the elemental nitrogen and the ammonia. Of course, in order to make and have a amino acid, you need nitrogen. So to deliver the nitrogen to make the amino acids, these guys definitely gave you a component, which is the end. So this is screaming to you the answer. Okay, so to provide, these guys provided the nitrogen that we needed to make the amino acids. They didn't talk about who provided the carbon. That would have to be the CO2 provided the carbon to make it as well. But they're saying, hey, just for the nitrogen part, these guys who have N, nitrogen, provided that element to build this compound. So that B is definitely the answer. Sulfur dioxide in the experiment in 2 could donate excited electrons required to drive the process of photosynthesis. Yeah, there are certain photosynthetic pathways where SO2 was used, okay? And that's not what this is about, though. Okay, and then D, methane gas could act as an electron acceptor required to complete the process of cellular respiration. I know there's something called methogenesis, where methane is produced as opposed to water, CH4, and they call it methanogenesis. But I don't know if there is an example of methane as electron acceptor. I know that carbon and hydrogen aren't very electronegative, so I'm unaware that this is exactly how this normally occurs. Normally in cell respiration, it's the oxygen, okay, that is very electronegative, attracts electrons and pulls the electrons through the electron transport system in oxidative phosphorylation. Methane, I don't believe, can do that. It doesn't really matter. C and D are talking about specific metabolic pathways in life. This is just talking about creating what? The building blocks. So C and D have nothing to do with just creating the building blocks. They're very specific types of things that the experiment was not testing for. So B is the best answer moving forward. You know, you have to always think to yourself, what's the easy solution to many of these problems? In number 23, we have this cladogram given to us. It's an evolutionary type of relationship, how the chimpanzee, human, gorilla, and horse divergently, most of the time, evolved. And looking at these positions here, why are they diverging from the chimpanzee? Now, of course, we could swing the human and chimpanzee and change that. Doesn't really matter the positions there, as we talked about. But what is the event here? Some event or some mutation caused the horse to be different from the gorilla. Okay, so Y, X, and Z represent positions here, or you can represent basically the type of allele that is different. Okay, what's the allele to else that is different here? What makes the horse and gorilla? They are related, but some event makes them different. Now, if you look at the amino acid at position 104, so it's I guess the 104th amino acid in a beta hemoglobin protein. This is a tertiary structure of a folded protein, okay? And they're folded because of how the R groups are attracting each other in the amino acids that are put together, and the amino acids that are put together. So these amino acids are bonded to the R groups. The R groups are attracting dependent upon what type of R groups they are. And at the amino acid position of 104, clearly there's a huge difference. The gorilla has something that the horse and chimpanzee and the human does not, and they have a different amino acid at this position. All right, so the validity of the cladogram, which by the way is a hypothesis based upon some type of evidence of alleles. In this case, we have some really, really strong evidence by DNA sequencing, or at least protein analysis of the type of hemoglobin being made. Any case, the validity of the cladogram best supported by the molecular evidence, okay, of the protein, for which of the following changes in the amino acid composition of the beta hemoglobin protein during the evolution of these species. So the only difference here is leucine. So the gorilla's got leucine while the others don't. So if I look at my cladogram, the gorilla, the difference here, sorry, the difference here is that the gorilla, okay, has leucine at that position in that protein while the others don't. So the event that occurred here that makes this guy different, okay, is that it has a leucine in its protein structure and the 104th amino acid of that protein, which makes it maybe have a little bit of a shape difference. Okay, now that doesn't mean that the horse, human, and chimpanzee don't have other differences. This is just one difference in one protein. What's making the gorilla move this way is it has something the others don't. Okay, if they both had the same, if the human had it, or the horse had it, or the chimpanzee had it, okay, they would basically be branched together. Okay, but these are different species. Okay, and there's going to be many more differences besides that one in that position, but that's just one of the differences. So back to the question: the validity of the cladogram, best supported by the molecular evidence, blah blah blah, okay, is equal to what? Well, arginine goes to leucine at position X in the cladogram. Well, we just said we now know from this piece of evidence what makes the gorilla different is that it has what? Leucine. So the rest of them have arginine, so arginine I should say. So arginine went to leucine for the gorilla at position X. There's nothing more to it than that. Take it when you can. So the answer is arginine to leucine at position X. What made the gorilla differentiate, okay, diverge, okay, from the horse or the other animals here, is that they had that change in its hemoglobin. Of course, there's a heck of a lot more other genes that are different, and we can't identify what W, Y, and Z are for the others, but we do know that the gorilla is different because of that, and that may lead to a different ability of the hemoglobin to work in different ways. We don't know anything about these other differences. We just know one. So X had to be arginine going to leucine. All right, and that little difference is enough for us to answer the question. And there's no more to it than that. It feels like this question should have more to it, all right, but it's that simple. So number 23 is A. Don't overthink some of these questions, all right. Number 24. Number 24 and 27, another cluster question. You got all this information, okay? And again, what I suggest is scan. So diapause is an interruption of an organism's life cycle. Interruption, stop, that kind of stops their life cycle a little bit in response to environmental cues or stress. The soil nematode is capable of entering an adult reproductive diapause. Pause, or pausing something when food is scarce. Okay, so what the heck does that mean? In this, individual become normally reproductive in 2 days after hatching and remain fertile for 18 days. They produce by self-fertilization. Okay, so the investigations, they examine the survival and reproductive success following times of ARD. The idea here is that you have to figure out that something is pausing. What's pausing is their life cycle when there's no food. That's the stress. So it seems like they're able to, when somehow kind of stop their life cycle, which normally is being mature to, they're mature for 2 days, and they mature after 2 days of growth, and they remain fertile for 18 days. Okay, well, look at here. We're looking at 30 days of no food, yet we have the brood side, which means the size of the amount of offspring. Okay, if you didn't know what a brood size, you'd have to keep reading. But the point is, we have some kind of pause in the life cycle when there's a stress or a lack of food. So let's go to the question. Don't spend any more time on this, and then go back if we need to. And number 24, which of the following best describes the reproductive ability of the nematode following the ARD induced in the experiments? The pause, the diapause, meaning we're stopping the life cycle because there's no food. So when we look over, you're going to have to go back to the evidence now. What's best describes? Well, let's look at the choices real fast. Mating with well-fed male consistently produced more offspring than reproduction of via self-fertilization. The numbers of progeny produced by self-fertilization are not statistically different. The nematode stopped producing after 20 days. This, my friends, if you can't realize it, is just a question of, can you read the graph? Can you interpret the graph? So we got to go back and check. So let's go through A. Mating with well-fed male consistently produced more offspring than reproduction via self-fertilization. We've got to go to the graph. We don't know this. So looking at our graph, we're talking about two graphs. They talked about self-fertilization and mating. So the average brood size, and again, if you didn't know the brood size, okay, the brood sizes you'd have to look up represent the what? The average number of offspring per adult. So that's how many children they could have. So what do we see? The size of the litter, or the size of the number of individuals that can mature, okay, is much greater when we mated, okay, the what? Mated the nematode with well-fed males, okay? And again, if you don't know that, there it goes. Well-fed males are the shaded bars. And so when we took this nematode and took away its food so that it paused its life cycle, okay, and then we mated it after 0 days, 3 days, 5 days. As the longer that the females were in this pause because there was no food, the lower what? Clearly there's some kind of statistical relationship. The lower the number of individuals that were born, okay, or offspring. But notice the mated males were statistically, if you took the starving females that were in the ARD and you mated them with the well-fed males, okay, you had a lot more offspring with the mated males than if the females basically fertilized themselves or had asexual reproduction. That's what that is. So go back to the question. If you get lost, reread the question, guys. Refocus all the time. Which of the following best describes the reproductive ability, okay, following the ARD induced in the first experiment, the first graph? Mating with well-fed males consistently produced more offspring than reproduction of self-fertilization. Well, that's what we're looking at. The shaded areas are bigger, and they're statistically bigger. Look at the error bars. These error bars are plus or minus 2 standard error of the mean. This gives us reliability. This gives us that the values they got are within the true mean of the population. So remember, this value here was read at, let's say, 375, but the confidence is in this area. But still, if you make this dot bigger, it's still statistically, because the error bars don't overlap, bigger. So all of these are higher, so that jumps out to be the answer, and you really should just move on. Don't spend time, but I'll explain why the other answers are wrong, only because you may have gotten those. The numbers of progeny produced by self-fertilization and mating were not statistically significant. Party people, yes they are, because the error bars are not overlapping. Remember, this is a 95% confidence range. And statisticians love 95% confidence. This is plus or minus what? 2 standard deviations of the actual mean of the population. So 2 standard deviation gives you that you're within all the natural variance of the population. Okay, 95% of all the variance. Remember, we have people that are close to the mean, and like me with height, we're farther away from the mean. So basically, by saying 95% confidence, we're saying that we're in this area of population, and this is what we accept to be normal or a good statistic. When we go beyond 2 standard deviations of the true mean, that's when we don't have a really good feel and confidence. So small error bars, they don't overlap. These are all statistically higher, every one of these. So mating, the nematode, starving female who is in ARD with a well-fed male always produced more offspring. That is the crowning achievement here, okay? But, so that's why these were not statistically significant. They were the difference between the self-fertilization and the well-fed males' sexual reproduction was statistically. They stopped reproducing after 20 days without food. So the starving females stopped producing? They still produced way after 20. Look at this. They still produce something, even if, of course, it's going down after 30 days with this break. Okay, they're still producing something. So that does not hold up. And then D, there was no relation between days without food and average brood size. I would say that you were wrong there if you chose that, because as the days that you starved them and then bred them, you can see that the longer you wait, the worse off it gets in terms of the size of the offspring. So there's definitely a statistical relationship there, and that's why D is not the right answer. Now, I'm going through the wrong choices because I want you to understand if you picked B, C, and D, why that wasn't a good choice. So 24 is A. Moving on to 25. The average brood size per mated individual upon reintroduction of food following 30 days of ARD is closest to which of the following? So the average brood size per mated individual upon reintroduction after 30 days of ARD is closest to which? Well, again, they just want you to read from the graph. So I'm going to go to the graph, and I'm going to try to find the value. So at 30 days, the mated individual, and I don't have a straight line, but I'm just going to read this top line and try to find where that goes. And it looks to me, again, hard to draw a straight line, that I'm somewhere near 50. All right, or 50 as they like to say. So I would say it's closest to 50. I'm just reading, like in my eyeball, and I'm just reading that line. So the mated value right there. So that's where they're getting that from, and that's why 25 is B. Number 26. So number 26 and 27 are the last 2 questions on this cluster of questions using the data above, and or the data that they give you. And what I will say about the last couple of questions, normally they are more challenging than the first couple of questions in this cluster. They're more interpretive. But if you're going to skip any and go back, it'd be the last question, the last 2. The first ones we can see are just basically reading right off the graph if you went over them with me. So number 26, what's the following conclusions? Now this is not just reading data now. Okay, conclusions are the leap that we talked about in our writing up our lab. Okay, what's the leap here? To take, what does the data mean? Okay, so this 24 and 25 was just reading the data. Okay, interpreting, or I wouldn't even say interpreting, they just reading off the graph. Now we're talking about interpretation. So 26 and 27, a little harder. So which of the following conclusions most consistent shown in figure 2? Well, if we go to figure 2 real fast, we can see that survival of the starving females that are in the ARD, and that's the diapause, that's the pause of their life cycle due to stress. And as the longer they are in ARD, which means they starve them, and eventually at this day they give them food, Okay, do they survive? And if you look, yeah, it's up and down, but carefully we have standard error of the mean plus or minus 2, which we should know quickly is nothing more than a 95% confidence that the actual number that we get from our data is within 95% of what the actual mean is. As I've drawn before in other problems, we have a range of possible means based on different sampling. And if we are within 2 standard deviations of the true mean, we call that a 95% confidence that you have your actual mean is within your calculated mean of your sample. And if the error bars are small enough, then you can be very confident in your number compared to others. Now, because these error bars overlap, what this means is that all of these values are within 95% of each other. And what I like to draw for people who don't understand is the actual dot here of confidence is right here. So if you think about it, I'm drawing these dots, I'm reading off to the right. We can't be confident that this number right here is, let's say, 17.5. We can only be confident that's between 21 and, let's say, 14. That's where the actual mean is. So because it's a huge error bar and this one overlaps, there is a great chance, a very good chance, that all of these actual numbers could be the same. There is no statistical difference, even though the lines are going up and down, because of the size of the error bars, which is usually tied to a low sample number. Okay, then these could typically be all the same what? All be the same size. Okay, now we did see that 30, okay, and 15, okay, are also big. So the idea is that there's a possibility that all of these are exactly the same size, because that line I just drew goes through every single error bar just about. So there's a great chance that there's no difference in these values, because the true mean was within 95% of all of them. Big error bars. If they're overlapping, no statistical difference. Okay, there's a great chance that all of these have a true mean that's exactly the same. Okay, and based upon the what? Poor sampling that you took gives you those differences. The samples were bigger, these error bars would get smaller. So back to so we're seeing that there's no statistical difference between how long we starve the nematodes and their survival. Meaning after 30 days of starving, then we give them food, there's basically statistically just as much survival as not starving them. Okay, and that's the important interpretation here. So animals that spent 3 to 5 days in ARD are more likely to survive? No, there's no statistical difference that there's better survival rates. So we get rid of A. Animals that spend 30 days live significantly longer? No, there's no significant difference based on the big overlapping error bars. D, the large standard errors of the mean make conclusions from this data impossible. No, it doesn't. We can still make conclusions based on error bars, right? It just means that our conclusion isn't going to have a lot of validity in terms of the outcome, or it could tell us that the conclusion, the big error bars that we're looking at could give us a conclusion that's pretty solid in stating that there is no difference in survival rates of different females being starved different days. So even though the reliability of the data is weak, given that reliability data, the standard of the means, we can conclude there is no significance. And therefore, we can make a conclusion that there is no real factor in terms of survival rates with being at different times of ARD or starving. So we can make conclusions based on error bars, okay, as we've done so in our labs. All right, so the best answer is choice C, has to be by process of elimination. Let's just read it anyway. The number of days an animal spends in ARD does not significantly affect its time of survival, and that's what the error bars are telling us. That should be our conclusion, okay? So 26 is C, okay? Okay, so number 27, this is the last question in a cluster. Normally the hardest question, the one that people get wrong the most. It's an interpretive question based on the data. Okay, in the other cluster questions, we had the first two, which were 24 and 25. We just read basically off the graphs. 26 was a leap, okay, from the conclusions that we talked about, and now 27 is the hardest question, an interpretive one that we have to really be able to dig deep in our understanding of why the data makes any sense, and that's why I made you write your conclusion. So based on the experimental results, which of the following best evolutionary explanation? So we're looking for the best evolutionary explanation. All right, and that's important for the occurrence of this ARD in this nematode. Of course, the ARD is this diapause, the pause in the life cycle of the nematode where food is scarce. Okay, they can actually starve and go into some kind of like little bit of a hibernation, and then of course giving them some food, okay, they can survive again, but they can also kind of shut down their metabolic pathways enough to still be able to reproduce if mated with themselves, asexually or sexual reproduction. Any case, so we're looking for the best evolutionary explanation, and this is tough because in this question we're going to look at distractors that are very similar, and we have to pick the best. Okay, and again, this is one of the questions where they're trying to get most people to get wrong. They don't want everyone to get hundreds on these tests. So look at the choices. The ability to enter ARD, this little hibernation type of scenario, okay, provides a strong selective advantage because reproduction can occur despite periods of food scarcity. Absolutely one that jumps at me as a strong possibility. If the organism has the genetic makeup to enter ARD because of a mutation or whatever genetic diversity it has, it's a strong advantage because when there isn't any food, the ones that do not enter ARD will die. So of course it causes to be a strong selected advantage. The only ones that survive and reproduce are the ones that have the genes to go into ARD when there is no food. Okay, so reproduction can occur when there's a low food available. So this one jumps at me as a strong possibility, but let's read the others. Acquiring the genes for ARD gives individuals a selective advantage. Boy, that is true. So far that's true, because they produce more offspring than individuals who do not enter ARD. This sounds like it should be true, but careful, because they produce more offspring than individuals who cannot enter ARD. Is that true? Do they produce more individuals? Look at 0 days. At 0 days, that means there's no starving, there's no ARD. We had the highest values for either self-fertilized or mated. Once you got into ARD, which is where? Where does the ARD start? Okay, the ARD starts here. From 3 days on, you can see there's a significantly lowered number of offspring. So that is not true, and that's a very nasty distractor, because you want to say, hey, I have a selected advantage because clearly there's reproductive fitness, and reproductive fitness, if you're a robot, means that, hey, you must make more offspring. In truth, they make less offspring, okay, than individuals who are not. But if there is no food, the ones that don't will die. So this is not true. This last statement right here is not true. They do not produce more offspring. And so the number of offspring is not greater. There's less of them. Okay, but the first part is true. So that's why this is wrong for B, but one that people pick all the time. Okay, do not confuse the fact that having a selected advantage always means that you produce more offspring. Basically, the selective advantage is that you can actually survive the low food times or low food periods, that are periods where there's no food available to the organism, and therefore the ones that don't have ARD just die. So you do have increased advantage in selective pressure to have ARD because you actually can reproduce when there's food. It's not about having more, okay? In this case, individuals who enter ARD are selected for in the population because they live longer. If you look at the data, do they live longer if you're in ARD? Look here. If you have 0 ARD, 0 days in ARD, statistically, because what? The error bars between having 0 ARD and what? And being in ARD for up to 30 days, there's no statistical difference. To draw a line right through, there's no statistical, I can't even draw a line, right through, there's no statistical difference. So that's not a true statement based on the data, so that's not supported by that, so we get rid of that. And then D, and again, going through the choice is important here because they want the best. Individuals who enter ARD have high fitness because they reproduce even when food is scarce. And people like this one too, and I like it too. Individuals who enter ARD have high fitness. Absolutely. And fitness is speaking to reproductive ability. Okay, so obviously they can still reproduce even when food is scarce, whereas the ones who don't enter ARD will not have the same high fitness because they will die, okay, before they reproduce. So D is a possible. But when we talk about evolutionary explanation, is this the best explanation? Which one's better? Reread them. The ability to enter provides a strong selective advantage. Hello, natural selection works because of a selective pressure. The pressure is very simply the ability to reproduce, okay, and basically live when there's no food, okay? So both of these are true, but I like talking about natural selection in terms of selective pressures, okay? So this is the best answer. There are two answers here, but the best one that speaks to evolutionary explanation is talking about a selective pressure. This is true, but they're not talking about the selective pressure. And when you talk about selective pressure, you are in fact bringing up or inferring natural selection, where this one isn't. It's talking about the ability to reproduce, which is correctly tied to natural selection too, but this selective, strong selective pressure speaks more to a better explanation. So A is better than D. And I know that's nasty. I get that. All right. So 27 is A. Number 28. Number 28, steroid hormones such as testosterone pass through the plasma membrane and bind to an intracellular protein as shown in the diagram below. The hormone receptor complex then enters the nucleus where it interacts with DNA to promote the transcription of a specific gene. Okay, this is the what? This is the reception, transduction, and response, the three steps in cell-to-cell signaling. So the reception, okay, oftentimes is a receptor or some kind of usually some type of protein in the plasma membrane that can accept some receptor that accepts the steroid, which could be a protein, in this case a hormone. In this case, because this is a non-polar small hormone, testosterone, it's able to pass right through the mostly what? Hydrophobic middle layer of our plasma membrane. It has those hydrophobic tails. So the reception isn't occurring in a receptor in this case, but there is a receptor complex inside. And if they didn't have that receptor complex, the next step wouldn't occur. So reception doesn't always occur by proteins that are attached to or glycoproteins attached to integral proteins in the plasma membrane. They can actually pass through, but you still have to have a receptor. Then of course the next step is transduction, and you need to know this. And transduction is part of the cell signaling pathway where we have a cascade of events that can occur after which. And then, of course, there's the response. And the response normally means somehow this enters the nucleus where it can be a transcription factor to turn on or off a gene and transcribe a gene to make a certain messenger RNA to make whatever you need to make in terms of an enzyme or a protein. So these are the three steps, and they're just kind of going over that with you. And then here comes the question. Based on the information presented, which is basically a cell signaling pathway of the three steps I just mentioned, which of the following will also occur? Now, what they're showing you is, of course, the reception part, and then transduction, basically probably the same thing happening at the same time, and then part of the response, meaning we're showing that there is a response and that this is causing a messenger RNA to be made, and then of course out of the out of the nucleus in the endoplasmic reticulum of some proteins being made. So What else? Look at the choices. Histone protein synthesis will occur. Now we know that we're making proteins here, that's for sure. Okay, we're going into the endoplasmic reticulum, and the two subunits of the ribosomes, okay, and tRNA, as we've talked about, has this anticodon here that codes for a particular amino acid that's being brought in, and that's happening here. We're building the amino acid chain. So you may say, okay, protein synthesis is a good thing. I forgot what histones are. Well, histones are proteins that keep a DNA condensed into such a condensed form that it can be separated in mitosis or meiosis. This condensed form, histone proteins, are basically N-methyl groups, okay, are there to keep this structure. And this structure of the DNA in a chromosome that's condensed like this is not open for transcription. We want our DNA to be open so that we can get the DNA polymerases in there to make the messenger RNA. In this form, in a chromosomal form, which we should remember only occurs, okay, late stages of the growth cycle, or a small stage of the growth cycle, that where we have this condensing of course this occurs only in mitosis, and prophase is the first place where it starts to condense. We actually break these histone proteins. We have enzymes that come in here and break these proteins down, or I'm sorry, we have enzymes that cause the formation of these proteins to condense the elongated DNA that's in this form, so we can use it to make proteins into a form where we condense it. So histone protein synthesis would be what we want to do when we want to condense and make this into a form so that we can separate it from each other. Remember, the elongated DNA strands is what we need to use them. So most of the life cycle of the cell is in this form. So this is not a good choice. Think about it. Histone protein synthesis will increase because histones maintain DNA in an optimal conformation for chromosomal assembly. That's true. Okay, it's true in that histones do maintain DNA. It keeps them in this area. Now, why do we want them condensed? Because when we separate chromosomes from each other, we want them condensed easily. If they were in this elongated, non-condensed shape, okay, other chromosomes might attach to them, and there could be all kinds of disastrous effects in mitosis. We want to condense up all the DNA into a chromosome and then have them separate into gametes or duplication. Before, this would be a mess of DNA if they didn't. So histone proteins help connect DNA into this condensed form. So that's not what we're talking about here. We're talking about where we're going to make protein, but not protein to condense the form, so that we start mitosis. Okay, this is prophase, okay, or meiosis. All right, so that's not the answer there. Ribosomal production will increase because ribosomes are specific for messenger RNA. No, they're not. Ribosomes are basically these subunits, and there's two of them there, that are able to take any messenger strand. Okay, now there probably are some ribosomes that are specific, but generally speaking, these ribosomes are sitting there. What's really specific is the tRNA. Remember, the tRNA have a specific amino acid based upon a specific anticode that will match the messenger RNA. Okay, so this helps facilitate the tRNA to come together. So the ribosomes aren't specific. It's the transfer RNAs are with the messenger RNA to build a particular amino acid strand. Okay, so they get rid of that. C, DNA replication will increase. Well, we're not talking about a growth factor here. We're just talking about making proteins. Now, these proteins could be a growth factor for its own cell, but generally speaking, if this was a growth factor, maybe that would signal to proteins that would do that. So maybe that is a possibility. But think about this again. DNA replication will increase as a result of binding of the hormone receptor complex to the DNA. Okay, really what we're doing here, and it's important you understand that when we bind the complex to the DNA, it is oftentimes a transcription factor that turns it on so that the DNA polymerase can make a messenger RNA to make a protein. So maybe that protein could be a growth factor that stimulates, but directly this is a tempting one, but it's not about DNA replication. It's about what's happening here. It's about making messenger RNA. So it's not about a replication of DNA. It's about transcription of a code for a protein to do something. Now that protein could lead, okay, to an increased rate of mitosis, absolutely, but directly speaking, it's about transcription, not DNA replication directly, okay? So that's a little bit of a tricky one there. And D, production of a specific messenger RNA, that's what it's about. When we talk about the response is getting in the nucleus and transcribing messenger RNA, that code for a certain amino acid sequence to make a certain protein that will do something. That's what this is about. So that's what the answer is, clearly D, the production of a specific amino acid sequence for a specific protein that specifically will catalyze a certain reaction, okay? And that's what this is about. So the choice is D for number 28. So number 29. Number 29. Okay, so we've got a pedigree diagram, and the first thing I want to talk about, what kind of pedigree diagram are we talking about? Well, they kind of gave it away, and sometimes they don't. They called it a inheritance of a dominant allele. Shows the inheritance of a dominant allele. So this is a dominant type of inheritance, meaning if you have the dominant A, if it's given to you, if you have it, that means you have the trait. Okay, so that's what this is really. Over several generations, circles represent females and squares shaded into carrying just the allele. So a dominant allele, you just have the A, which means you have the disorder if you just have one of the alleles. Okay, and so 50% of your offspring have it, as you can see. Generally speaking, it's a 50% trait. So you just have to have the allele. Case in point, a great example is Huntington's disease. Okay, if you just have one of the alleles, okay, you have the disease, unfortunately. And it's not a sex-linked trait whatsoever, as you can see, male or females, but it's about 50% of your offspring, okay, have the chance. So that's why you see it proliferate between every generation. Okay, any case, the pedigree suggests that a gene is on a nuclear chromosome, not on a mitochondrial DNA. Because why? Well, we have to understand something about embryology that we talked about. We have these follicle. Okay, remember there's different cells around the different types of cells that surround the female egg, let's say in a human embryology. And of course, these different cells help produce different hormones that keep the balance of hormones correct in the female, so that you have the right type of endometrial lining, so that there is a pregnancy or implantation of this fertilized egg. And then of course you have the sperm, who carries the DNA. Okay, and of course, because the sperm has to travel a far distance up the fallopian tube, okay, through the uterus to the fallopian tube, and eventually to the ovaries, where these follicles or living eggs live. Okay, the sperm doesn't carry a huge amount of cytoplasm, and we talk about this because there's cytoplasmic determinants already existing in the cytoplasm, big cytoplasm of the egg. Now I should draw the nucleus a little bigger, okay? But there's a huge cytoplasm, and then of course how these determinants arrange themselves in the first couple of divisions, once there is a zygote developed by the union of the sperm and the egg to haploids to a diploid cell, determines the types of direction and axial, and where the head and where the foot is, and determines what type of cells are made, generally speaking. But what I'm talking about is the sperm doesn't have a lot of cytoplasm. In fact, there's very little of it. Of course, it does have mitochondria, but what we need to understand is the mitochondria come from the female. And as we know, mitochondria have their own DNA, and I'm trying to draw mitochondria here as that cystae, those in a membrane I'm trying to draw. So really, we get our mitochondria, which we know that we've gotten evolutionary from some other engulfing of a prokaryote or a eukaryote, engulfing this prokaryote once by endosymbiosis, because the DNA of the mitochondria is unique to its own. It's a organelle that has its own DNA. But the bottom line is the cytoplasm, once this cell starts to divide, okay, and make two daughter cells, once there's fertilization, there's a division of the cytoplasm. And so because the DNA of the sperm goes through the nucleus membrane, if it fertilizes and then eventually gets into the nuclear pore, that's where the union of the male's DNA with the female's combine to make a fertilized zygote, a diploid cell. But it's the division of the cytoplasm that results in dividing the mitochondria up. And the mitochondrial DNA, okay, or the mitochondria come from the female, okay, not the male. The male doesn't bring any. It just brings the DNA. So the cytoplasm of the egg. So our mitochondria that we have always come from our mothers. So the pedigree suggests that the gene is on a nuclear chromosome, not mitochondrial, because the mitochondria genes are not heritable. Well, mitochondrial genes are heritable in the sense that we get our mitochondrial DNA from our mother, so they are heritable. They just don't come from the father. Okay, mitochondrial mutations cannot produce dominant traits. Yes, they do, because since we get our mitochondria only from our mothers, then if that mitochondria, remember, mitochondria do self-replicate on their own, even though they're controlled by the nucleus somewhat. They have their own DNA to duplicate themselves. It makes the whole process of duplication of mitochondria more efficient and fast. Okay, so if you only have one type of mitochondrial DNA because you only get the one from the mother, they do produce dominant traits. If you have a problem or a mutation in your mitochondrial DNA, that'll show up. There's no other allele from your, let's say, father. You don't get mitochondria DNA from your father. So essentially, these are dominant traits always. And C, maternal mitochondrial mutations are inherited by all the mother's offspring. Correct. So this shows you that this is a nuclear, not a mitochondrial, because this would be an A. This would every single offspring would have, I should say, all the offspring, I shouldn't fill this in yet because we don't know based on the father, but all the offspring from the mother. Okay, so because this one is empty, it's showing that it's not mitochondria. Remember, the mother gives all of its offspring its mitochondrial DNA or mitochondria. We get our mitochondria from our mothers 100% of the time. So this would be filled in. So therefore, maternal mitochondrial mutations are inherited by all the mother's offspring. That is correct. That's the right answer. And then why can we eliminate D? Mitochondrial DNA is circular. Yes, it is, because it's just like a plasmid of a prokaryote, whereas chromosomal is linear. This is true, but that's not an answer based upon the pedigree chart. The pedigree chart is showing that some of the offspring do not have the trait, and that would have to be a nuclear chromosome, because mitochondrial DNA or mitochondria all come from the parent, and every single offspring would have it. So that's why we get rid of D. D is true, but it doesn't answer the question based upon the pedigree diagram. So C is the best answer for number 29. Number 30. Okay, so number 30 is that moral, ethical type questioning that we get with genetic results. So high blood pressure can lead to cardiovascular problems, as like heart attacks. Atherosclerosis is the hardening of the arteries that make it hard for the heart to get oxygen and may possibly die. That's what a heart attack is. Exercise and diet can often control the cholesterol and limit the hardening of the arteries, or the closing of the arteries, which is really what atherosclerosis is. When the arteries start getting closed, there's lower amounts of oxygen that get to the heart muscle that contracts our heart and moves the blood. If this closes off more, that's what heart attack is, that there's death of the heart cells. Okay, now having high blood pressure is a heritable trait. It's caused by an autosomal. Autosomal is a non-sex chromosome, and it's a dominant disease. Okay, autosomal dominant meaning it's based upon having that gene caused by the mutation of a single gene. Okay, a man with a high cholesterol levels is about to marry a woman whose total cholesterol levels are also higher than average. A physician has suggested they get tested for the HC allele. meaning do they have a genetic factor beyond their diets that's causing high blood pressure, which is a risk for heart attacks and death and more medical bills. Which of the following is a valid ethical question concerning the test? Now, when you hear ethical, we think of a moral dilemma. Moral is the right and wrong of the issue. Okay, so let's say a question that's really judging what is a right and wrong type of question, which again is based upon the answers for the individual. Okay, how important are the results of the test to the health, since they already know they have it? This is not a moral dilemma, okay, whether or not the results of the test are important, okay? So this is not an ethical question. How could they eliminate the allele? Again, that's a question of science, or that we can't just eliminate alleles. So that's not a moral dilemma, okay? And then D, what cures are available for high blood pressure or for this gene? That's not a moral dilemma. It's a question for a Winkin science. So you should be able to eliminate your wrong choices and see that the process of elimination sees the answer. If both have the allele, should an insurance company raise their rates because of the results of the test? This is a moral dilemma. Okay, even though it's one for the company, what's right and wrong for the company now that you know that these people have the allele, have a higher propensity for the disease? Should they raise their rates now knowing their genetic makeup? And maybe they should, because other people paying the same insurance, okay, is based upon, okay, the risk. If you think about how insurance works, everyone pays a premium, which is basically a little bit of money into a pool of money that people pull out when they have problems. Well, if people have a higher risk, then shouldn't they be paying more money into that pool? Because they're going to pull more money out if they get sick or have a higher risk of sickness. This is a right and wrong moral dilemma, okay, for the insurance company. Probably a moral dilemma is that should they report their findings to the insurance company? Okay, what's the right and wrong of this? C is the best answer there. Okay, so number 31. 31, a cell is treated with a drug that prevents the formation of new lysosomes. Lysosomes are those packaged vacuoles that have enzymes that have digestive properties. The cell continues to transcribe the genes that code for the hydrolytic enzyme. So therefore, transcription isn't stopped. Okay, the drug doesn't stop a transcription factor from going into the nucleus to write a primary transcript from the DNA. Okay, and continues to translate, so the drug doesn't stop the translation. So basically, we're making the protein still. We're transcribing it and we're translating it into proteins. The hydrolytic enzymes are most likely to accumulate where? So we have a drug that stops the prevention of the lysosomes. So we're transcribing it, so we're making messenger RNA. We're translating it, so therefore we're making protein. We're making the enzymes. So if we're not making them into the packaged vesicles that we call lysosomes, where is this going to stop? Well, it's not going to stop the nucleus because the messenger RNA is made in the nucleus. It's not going to stop in the smooth endoplasmic reticulum because, well, if we're going to make a protein, there's going to be ribosomes in our smooth endoplasmic reticulum. Understand, the endoplasmic reticulum is basically a passageway, a tubular passageway for messenger RNA to travel from the nucleus, where the DNA is only, and moves to the endoplasmic reticulum, but it'd be the rough endoplasmic reticulum that has the subunits, the ribosomes, okay, that makes it rough to make the proteins. And then the proteins are then moved to the Golgi apparatus, where they're packaged and basically sent to areas specific. So the packaging of these proteins, hello, the vesicle is a packaging of protein for a certain location, has happened in the Golgi complex. and that's why 31 is D. Now, B is the mitochondrion. That's, of course, the energy organelle that we've gotten from endosymbiosis. That has nothing to do with making enzymes, although it has something to do with making or helping couple the energy from a glucose to make ATP to help with the production. It directly isn't involved. So the Golgi apparatus is what we're talking about here. So 31 is D. 32. So number 32, the 32, we have a diagram above, and in our diagram, we see the following. We have DNA damage. This damage creates some kind of internal signaling communication of protein kinase cascade, which creates eventually P53 phosphorylation, okay, which acts as a transcription factor that goes back on the DNA, and that DNA makes a protein, and that protein somehow inactivates the cyclin CDK complex. Okay, so what do we have with the question? Well, the P53 protein regulates a cellular response to DNA damage. The damage created a internal cell signaling pathway, a transduction pathway that created other proteins to affect another cyclin CDK complex. Okay, so the response of the DNA being damaged really created a scenario where we phosphorylated another internal protein that acted as a transcription factor to make another protein, which basically suppressed and inactivated another protein that was in the cytoplasm. That's what's happening here. So if you look at your choices, which of the following best describes the role of P53 in response to DNA damage? We can see that the P53 binds to the DNA and repairs the damage. That's not what's happening. The P53 that's phosphorylated binds to the DNA that transcribes a new messenger RNA that gets translated into a new protein. We're not transcribing from the DNA damage. That would be a bad thing. If we're making messenger RNA from this place, we'd create proteins that won't work, okay, and that would be very disastrous. So that's why we get rid of A. Now, choice B, phosphorylated P53 stimulates the transcription of P21. That's true. And this suppresses cell division until the DNA damage is repaired. Now, I don't know if it suppresses cell division, but if we remember our form, cyclin CDK complexes actually are part of the metabolic up and down cycle of activated cyclin, and this up and down activation of the cyclin CDK process, remember, is one of the factors that turns on the cell cycle from, let's say, a G1 to the G2. Now, if we look at the basic cell cycle, let's make this mitosis, and that's where new daughter cell is created. There's only a small part of the cell. Then we're going to have the G1, the regular growth phase, where the cell gets bigger and does its job, and some cells will cease dividing and die here. If they go on past the G1 phase, then they're into the S phase, and that's where we have duplication of the DNA. And then we're into the G2 phase, but we're in preparation for mitosis. And then, of course, we have the prophase, the metaphase, the anaphase, the telekinesis, all of that good parts of mitosis where the chromosomes are condensed. But the point is, there's different checkpoints that start and stop. Make sure that there is a multiple checkpoints in this cell cycle. Ensure that the division occurs only after sufficient growth and faithful DNA replication. All of these checkpoints ensure correct regulation of these steps, and therefore proper mitosis that carries on the organism's traits to new daughter cells, and that's the key to evolution entirely. So when you think about it, we need cells to divide and carry at least their own copies to mitosis. Now, evolution, of course, is genetic diversity, but it is actually the mutations that can occur from the duplication process in the S phase, where it leads to all of the genetic diversity that occurs. So it is tied to that. So we need to have careful checkpoints so that the daughter cells do not have the wrong number of chromosomes and can go on and live a nice happily cell life. And these checkpoints are dependent upon cyclin CDK complexes, or better yet, they're cyclin dependent kinases. What they are is you have proteins that have no enzymatic ability until they make a complex with a kinase, and then they're active. And there's different types of these complexes for the different checkpoints in the different parts of the growth cycles. As we go from G1 to S to G2 in mitosis, there's different checkpoints that different cyclins have to be in greater quantity. And they're cyclin because they'll go up and go down in the process. But the bottom line is, you really didn't need to know all of that, but that's a good review of the checkpoints in mitosis or the cell cycle. What you needed to know was that this DNA damage created a cellular response to make another protein that eventually inactivates, okay, this cyclin, which is going to stop mitosis. Now, if you didn't understand that cyclin does that, your choices would definitely lead you there. So going back to the choices, A repairs the damage. There's no damage being repaired here. We don't see that. There's the damage in our drawing, and this model is showing that the damage isn't taken care of. Somehow we're just inactivating this protein. That's all you know if you didn't know that cyclin was part of the growth cycle. And then look at C, phosphorylated P30 binds to the cyclin complex. It does. The P53 is binding. No, the P21 is binding. So this is just reading right off the graph. Okay, so this has got nothing to do with it. So, choice C is just reading or just looking at the graph to see that. So that's got nothing to do with it. And phosphorylated P35 inactivates P21 proteins. Does it? P53 inactivates P21? Well, in truth, P35 phosphorylated is a transcription factor that turns on the gene to eventually make P21. So you can think that P35 kind of, in some ways, activates, not really activates, it promotes and helps make P21. Okay, so maybe you could say it activates, and then the P20s in turn repair. So maybe this part we can consider true, definitely. And the P21 repair the DNA damage. How would you know this repairs the damage? Again, if you didn't know that the cyclin CDK complex is part of a checkpoint in the growth cycle of the plant, in terms of the cell, it could be a plant, repairs the damage. We don't see any repairing of the damage in our diagram, so we get rid of that. This is a classic case where the first part is true and the second part is there's no diagram to show that there. So we're left with B, okay, which states phosphorylated P35 stimulates transcription. I like that better. Okay, then activates anyway. And the resulting P21 suppresses cell division. Okay, since we have repairing the damage in A, repairing the DNA damage in C, and repairing the DNA damage in D, and you never see that in your diagram, you're just left with B, even if you didn't know the suppression of cell division was tied to CDK. So that was a total choice you could have got from eliminating the bad choices based upon the drawing above. 32 is B. Number 33. Questions 33 to 37, cluster questions. How do we attack these? Scan. Scan to see what information you think you have to know, and then go right to the questions. Now, why do you do that? Because the first couple of questions are the easiest in a cluster, and it's the last ones that take more time. So you want to get to these questions first. So I'm just going to go right to number 33. Which of the following graphs is the most appropriate representation of the experimental results? So I don't even understand what they're doing. Let's look at the results. So what graph is best represented? And we've seen this type of question before. So I see error bars, pie graphs. Okay, let's look what we see here. Well, I see, I see 6 treatment groups, okay, light, dark, okay, we're measuring, I guess, pH, and I see that there's 6 different, isn't like we took the same control group, or same group and changed things on them. We have 6 different groups and then 6 different averages, and then we have the standard error of the mean, which is what the error bars are built upon, right? That gives us confidence in the value, because standard error of the mean, you should know very easily at this point, is how close is your mean, okay, to the entire mean of the entire population in the universe, okay? With a low standard of the mean, that means you could say with confidence your number is pretty close to the actual, all right? So if you look 8 to 6 to 5 to 5 to 7 to 6.8, I'm just looking for data that represents this the best. And if I've got, in this case, 6 different control groups, or treatment groups, I say, that are different from each other, they cannot be interconnected like this or like this. Okay, if there's 6 different groups, I should show them separate bar graphs. And of course, I should have my error bars. Notice 5 has the biggest standard error of the mean, and does it? Just to make sure, it does. Okay, and so we're going starting high, going low, 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 and going up again, and then down again. And if you just look carefully, starting high, going low, 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 up again, down again, that's the same data. So this clearly is the answer. All right, pie graph would be if you had percentages reported, and there isn't percentages reported, okay? And if you have standard error of the mean, you have means, so this doesn't make any sense. And of course, C and D would never be a scenario where you have different treatment groups going up and down. A line would mean you have one thing changing over time. So if you can see that this is an easy one, you should grab it, 33. You shouldn't be spending 5 minutes to read and then get to B. The easiest questions are usually in a cluster, especially with graph and information and data given, are the first couple after. They're basically reading off the graph. See if we can find another one. Again, we haven't even read what the study's about yet. Which of the following observation provides the best evidence that photosynthesis occurred in treatment group 1? Okay, so now we're looking for observations, okay, that photosynthesis occurred. And so now we're going to have to go back and quickly read what this is about. So we have investigated a light and carbon cycle in aquatic systems, performed controlled experiments summarized below. They placed equal amounts of water that started with a pH of 7 from a large aquarium in glass beakers. They transferred aquatic plants. They placed equal number of beakers in light and dark. They transferred some goldfish. And they placed equal number of those in light and dark. They placed equal number of beakers with only water and dark. There's your controls. After exposing samples of light and dark for an hour, students recorded the pH changes. It started at 7, so they either went up, which means they got more basic, or they went down, they got more acidic. Now, what would do that? There has to be something in this. It's telling us that. Okay, well, here it tells carbon dioxide dissolved in water will lower the pH. That's the key. Ooh, we've seen this before. So CO2 going into water is going to lower the pH. Or if CO2 leaves the water, right, if you pull, so we've got this water. If CO2 goes into the water, the pH is going to decrease. They're telling you that. But if the CO2 leaves from the water, if you pull CO2 from the water, the pH will increase, right? You're taking away the acid. So CO2 in water is acidic. So you have to understand, by dissolved in water lowers the pH. So when you put CO2 into water, the pH drops. When you pull the CO2 out, the pH is going to rise. Okay, that's important. So high CO2 equals a pH that drops. A decrease in CO2 is going to give us a pH that increases. So what you needed to know is that basicity is higher pH and that acidity is lower pH, and CO2 is acting like an acid. But even in that, they don't have to know that. So they told you that little piece. Now, back to the question. Which of the following observations provides the best evidence that photosynthesis occurred in treatment group 1? Well, in treatment group 1, we have light with an aquatic plant in water. Now, what is photosynthesis? Photosynthesis is when you take CO2 and water, got plenty of water since it's sitting in water, and you use it, okay, to make some kind of carbohydrate, C6H12O6, plus oxygen. They didn't say what oxygen does, but we're going to use up the CO2 in the water as the reaction goes forward. photosynthesis uses CO2. That means the CO2 is going to drop. And if it drops, the pH is going to go up. So, does it go up? Does it go up? And, well, it's 8.2. How do I know it goes up? Because we read carefully that the pH started at 7 for all of them. So in all of these, they start from 7. So if we go from 7 pH to 8.2, the pH was rising. And that supports photosynthesis, because as they remove the CO2 from the water, as the reaction goes to the left and uses up these guys to make the carbohydrate and the oxygen in photosynthesis, we're going to use up the CO2 and the pH is going to rise, because adding CO2, the pH drops. So yes, let's see what the question states. This one required some little bit of effort. The specimens in the beakers were aquatic plants. That's nice, but that doesn't provide the observation that photosynthesis occurred. The beakers are placed in light. That still does not give us photosynthesis, even though light's required. The mean pH increased. Okay, that's the key. The pH increased after an hour. Remember, the pH started at 7 and now went to 8.2. Why? Because we were using up the CO2. The CO2 lowers pH. Take it away, it raises pH. That's why C is the best answer. That took some work, took some understanding. And I can see skipping that one if you're out of time. Okay, the standard of the mean just talks about the reliability, the confidence of the data, not the data itself. So 34 is C, 35. Number 35, to investigate whether an organism in the study is capable of both photosynthesis and respiration. Okay, a comparison of which treatment groups is most appropriate. Well, we just, in number 34, if you were with me with that, reviewing that question, we just realized that photosynthesis is going to use up the CO2. So the CO2 is going to be used up because we're going to fix the CO2 using Rubisco and add it to the carbon chain to make our carbohydrate or whatever carbon-based intermediate from G3P that we have. Any case, that's going to equal dropping the CO2, the pH is going to increase. And we saw that with 1. So because you have to use 1, it automatically makes the answer A. But why is treatment group 2 important? Well, if we have light on 1, okay, the light bulb or whatever is on and it's bright, the pH increased, as we just said, because we used up the CO2 in the water, and therefore the pH increases because CO2 in water drops the pH, so removing it raises the pH. Hey, and in 2, we have a plant in darkness. So we have a plant with light, where it's going to photosynthesize. In darkness, the plant still has to live and still needs energy, but it doesn't make any it's not using photosynthesis because there's no light to absorb. So what do we know in cellular respiration? Well, cellular respiration, we take the glucose that's already made, and for plants, this could be the starch that's already been made from photosynthesis earlier, that's stored, and with the oxygen, which is something that was made in photosynthesis, which is also dissolved in the water a little bit, you're going to produce CO2 and water. Now, I'm not balancing these reactions, but you should know that this is cellular respiration. So we're going to produce CO2 in cellular respiration as we cut up the carbons. Remember, plants have mitochondria. The energy they need to live is actually from the chemicals that they make through photosynthesis. So the idea that they make their own food is true, but they do have mitochondria and undergo cellular respiration. So they're going to make CO2 in this case. And notice, oh, the pH drop. Remember, the pH starts at 7, and this went to 6.4 at nighttime. So this tells me that they're photosynthesizing, okay, overnight. I'm sorry, overnight they're undergoing cellular respiration, okay, cellular respiration, because they're increasing the CO2. And if you increase the CO2, remember, it states that carbon dioxide dissolved in water will lower the pH. They all start at 7. Remember, right there, it says that they all started at 7. So dropping to 6.4 dark plant and water means that CO2 was being produced. And over here, we were taking the CO2. So 1 and 2 is the best answer. But you didn't have to go through all of that, but understand that that definitely proves it's cellular respiration. is occurring. And the only one there with 1 was choice A, so the rest of these make no sense. 35 is A. Number 36. So number 36. The results for the treatment groups 5 and 6 could suggest which of the following questions about the design of the experiment. So when we go back to 5 and 6, we see at 5 and 6 we have just a beaker of aquarium water in light, and then we have a beaker of aquarium water in darkness. And what do we see? We see the mean pH increasing a little bit, which we now know from the other question means that we lost CO2, and losing CO2 means that there was some possible photosynthesis occurring. Whereas in beaker darkness, which is 6, it's empty, we have the pH for 6, okay, decreasing. Now, why no decreasing? They both started at 7, and if it's decreasing, we're increasing the CO2. So there's possibly some cellular respiration going, because as you go forward in this reaction, you produce more of the CO2. Here, you're losing the CO2 as you go from left to right. So there's something possibly going on with our controls. There's possibly some kind of photosynthesis and cellular respiration that's occurring in these two beakers. All right, and let's look at the choices. The results of them could suggest, always good to reread the choice here, the question, to note, to refocus what you're trying to answer. So the results of for the treatment groups could suggest which of the following questions about the design experiment. Do the glass beakers prevent light? Preventing light has nothing to do with why one beaker that has a higher or lower pH. B, is the method used for measuring pH harmful? Again, nothing to do with it. We didn't talk about living or dying organisms here. Is the availability of carbon dioxide a limiting factor in aquatic ecosystems? Well, did we test different levels of carbon dioxide to see if we had different healthy or not healthy ecosystems? No. So by process of elimination, again, D has to be the answer. But here's a great choice. Does the aquarium water contain living microorganisms so small that we cannot see them with our naked eye? And therefore, these microorganisms could be photosynthesizers like algae, or they could be bacteria that are cellular cellular respirators, okay, or organisms that undergo cellular respiration. So the reason why we have beakers that look like nothing's in them, the fact that their pH changes could be, in fact, due to microorganisms that undergo cellular respiration in beaker 6 or photosynthesis in beaker 5. So this could be blue-green algae, this could be bacteria, it could be both. All right. In any case, that's a great little question there, but you could have got to the answers pretty quickly by process of elimination. So number 37. So 37, I would suspect to be a challenging question, one of the last questions in a cluster question, but it winds up being very easy if you understand something that you should know. Standard error of the mean, okay, is nothing given to us in our reference table is the standard deviation over the square root of n. Okay, so if my n becomes a larger number, that's the sample size. So if the denominator gets bigger, I know it's a square root, but if it gets bigger, the size of the standard error of the mean, okay, decreases. And what does that mean? If I take a sample size, remember, we're always taking sample sizes because we can never measure the entire population. We're trying to make a random sample and make a good sample. Remember, a good random sample of a population will give us all the different types of organisms that have a range of alleles or percentages of alleles. Remember the bell curve, bell distribution. So if we take a good random sample, our n will reflect, okay, the organisms, the actual true mean of the population, which is always after. We don't actually can get that. We're always estimating the true mean. But as our n number increases, okay, the standard error mean is going to drop, which means the range of numbers that we accept with very, very good high confidence becomes smaller. And if our range gets smaller, the error bars get smaller, we have a higher confidence that our number is an accurate representation of the true number in the actual population. So, which of the following modifications to the experimental design will help reduce the standard error of the mean? Well, if you want to reduce the standard of the mean, you need more uh, a larger sample size. The larger your sample size, the closer you're going to get to the true population. So increasing the sample size of each treatment group, okay, will definitely lower the standard of the mean, which would increase your confidence. Remember, if the standard of the mean is lower, that means your error bars are smaller and the range of high confidence becomes greater for a smaller distance. Okay, so C is the best answer. Really an easy question if you understand standard of the mean, which by the way, you have this formula in your reference table to look at that, and that could have helped. So 37 is C. Number 38. Okay, number 38, a good challenging question here. All right, so many cells can be stimulated by dividing by a hormone-like growth factors that, have this reception of a receptor by some factor that triggers a cascading event. But what's different here is we have these CAM, okay, these cell adhesion molecules that interact with other cells that have them. And when they bind, okay, they change the conformation or the structure of the protein so that this M molecule or complex will bind with it and it blocks the cascading events. All that means from this information they read is that when two cells are in contact with each other from these two CAMs, there's a conformational change of the protein so that the cascading cellular response that normally would happen with the three attaching onto an unlinked CAM would cause the RAS to link and also stimulate cell division. When you have the cell adhesion molecules attaching and changing the allosteric conformation of the proteins, the RAS can't bind, the 3 can't bind in the cascading events, and you have this blocking by the M and there's no cell division. So what this means is that this is a model that we understand that if we already have cells connecting to cells, there's no need for cell division because this cell is in its place, and this prevents a tumor, which is uncontrolled growth. Now, if a growth factor hits here and causes the reception, and then of course the cascading cell signaling pathway occurs, when the two cells are connected and this M is blocking because of the allosteric change, 3 can't go on here, and then RAS can't be stimulated to make cells divide. It prevents it, and it's a good little checkpoint or a control because we don't want out of control cells. If we're already next to a cell and we're just maintaining, let's say, liver cells, we don't want our liver to be growing uncontrollably. That's a tumor. We want to only replace cells that die. So when this cell undergoes cell apoptosis and you have no CAMs connected here, so let's say this cell dies. This one can actually undergo cell division because without the CAM there, okay, or without the other CAM hitting it, there is no allosteric change. And now if you have a growth factor, which would be released, okay, by many different avenues, it would cause a cascading event, which you could use the CAM as a enzyme at the end to make RAS eventually get activated, and cell division can occur to what? Replace the cell that was there and make a new CAM connection, and then there's going to be no more cell division because we replaced the cells that died in that area. So it's a nice little checkpoint. So when you think about all of that, and that's basically what I'm telling you from the reading here, you needed to understand the reading. This one required that. Which of the following statement accurately uses the information presented to support the hypothesis that an interruption of the M function, which is the blocking of this site for 3 so that RAS can't go, can result in cancer? Well, cancer is uncontrollable growth with no checkpoints. Let's go through our choices. Protein 3 will be prevented from interacting with CAMs. If protein 3 is prevented, then there will be no cell division. Protein 3 wants to interact with the CAM so that RAS somehow gets activated to make cell division. So preventing protein 3 would stop cell division. Okay, if you stop this cascade event, then the whole process stops. There's no cell division, there's no uncontrollable growth, there's no cancer. So A is tossed out. B, the RAS protein will remain bound to the DNA, blocking expression. Well, we don't know if it goes there, but the RAS protein will remain bound to the DNA, blocking the expression of genes required for mitosis. Now, those genes, of course, as we talked about in other problems, cyclin proteins. But if you block the genes required for mitosis, you don't have mitosis, you don't have uncontrollable growth, you don't have cancer, so we get rid of that choice as well. Growth factor signaling can trigger mitosis in cells that are in direct contact with other cells. Let's think about this. Growth factor signaling can trigger mitosis in cells that are in direct contact. So what tumors are is that you have direct contact, and somehow with a signal there's a bypass. There's some trouble with M blocking. So somehow the cancer is overtaking this. So that's my answer there, that I'm going to have a trigger of mitosis, even though I am in direct contact. So this M somehow is not blocking the pathway, and this cascading event is happening even though we are in direct contact. So somehow we're overriding this checkpoint, this blockage. That explains uncontrollable growth. And then D, if you're not sure, if you can cross it out, you know C is the right answer by, again, eliminating your distractors. The receptor proteins of body cells will no longer bind. Well, we're not talking about the lack of binding. We're talking about the system here, because they have a growth factor. It's again, this model is about how the CAMs are interacting. So, which of the following statement accurately uses the information presented above? This is about the CAMs, about being next to other cells and losing that checkpoint. So C is the best answer. Okay, that one probably takes some time to do, and you're gonna have plenty that are fast. And believe that if you take the questions that are fast and easy, you'll have time for some of these that do require a little more thought. But even if you don't get to it, it's still a single point question. All right, moving on. Number 39, we have a cross between a wild-type female Drosophila fruit fly and a mutant, and they give us the genotype. And when we do the cross, we get expected results. Notice these are all the same value, so this would be a 1 to 1 to 1. If so, you were to do the Punnett square, and I'm not going to do it, okay, you would get the same values. And when we do Punnett squares to get the expected, what we're talking about is Mendelian genetics. Now, Mendelian genetics is assuming a couple laws. The first one is called the law of segregation. Now, segregation is the idea that if you have a maternal, that's from your mother chromosome, and you have the other homologous chromosome, which is the same chromosome except that it's got your, let's say, father's, it's come from your father, and this chromosome came from your mother, and they have the same type of genes on there. Okay, now segregation means that when you make the gametes, you have a 50% chance of getting this chromosome or getting this chromosome in a gamete. All right, so obviously we talk about the offspring, okay, and the offspring, are you going to get one or the other in law of segregation? So your crosses that we get are depending upon basically a 50% chance of getting one chromosome the other. And that's the first law. Now the second law is independent assortment, that if you have two different alleles, okay, from two different chromosomes, then what you would get is the idea that if these chromosomes will independently assort from one another, meaning that getting, let's say, flower color, purple or white, okay, is going to be a trait that will be independent of whether the seed is round or elongated. Okay, so these are two traits, and so these are the two basic laws that come out of Mendelian genetics, because Gregory Mendel used pea plants, and he was lucky enough that the traits he was testing were not linked. And therefore, if they're not linked, the alleles are going to be on different chromosomes, or we'll learn, or they're really close together on the same chromosome. We'll talk about that in a second. Okay, or depending upon whether or not they are close together, we'll talk about the ability to be linked. So Mendelian genetics is about the fact that these alleles, these traits, these expression of the genes were essentially basically done by chance events, and nothing got in the way of those perfect Punnett squares, because he was lucky that the traits he tested were not linked. Okay, all right, which really means for most of his work was that the traits were on different chromosomes. Any case, so law of segregation, chromosomes will separate, or alleles will separate independent, will basically go to one side or the other. Independent assortment is when you have multiple alleles that have no linkage, meaning the chance of getting both of them are two independent events. But when you have a linked gene, that the number that you get in these crosses are not what you expect, and that's the case here, party people. Our observed phenotypes, oh, look at this. We have little b, dominant, I should say heterozygous dominant and heterozygous dominant for both traits for the female, and this is for homozygous recessive for the male, and it looks like most of the offspring has the parental genotype. And then the recombinants, now the recombinants are the new, okay, in this case, genotypes or phenotypes that we see, okay? And so these are the recombinants, and they're lower. And why are they lower than expected? Well, because of crossing over, and because these genes are linked. Okay, so they're giving you the expected, and this, put a big star next to this question, and the reason I'm saying a star, because they love to give you the phenotypical results, and then what you should get based on Mendelian genetics if the genes are not linked. But when they don't come out to exactly what we expect, then we have something called linked genes. Okay, which means these genes tend to travel together because of their linkage. Now, we used to think the linkage was they're on the same chromosome. So if you have this other allele on this one here, okay, make it black, and let's put it here. These genes travel together if a chromosome were to just segregate as Mendel suggested. Now, he didn't suggest any chromosomes, but he was talking about pure alleles. Didn't know how the mechanism was, but now we know it was chromosomes. Now, of course, crossing over can occur when you make gametes, and so if these guys are close enough together, okay, crossing over won't separate them. And so we'll talk about that in a second, but let's look at the choices. So this was a question, really, can you identify, party people, that the reason why the observed phenotypes are different than expected, that they're linked? It's got to be jumping at you. They're not coming out as expected. These genes are close together. They're traveling on the same chromosome or very close together on the same chromosome. So given all of that, let's look at the question. In cross diagram above, the expected and observed results are shown. Which of the following best explains the observed results in the cross? Well, alleles or body color and wing shape assort independently, as predicted by Mendel law. Well, that would be true if our observed phenotypes were very close to the expected. And remember, to know that they're different statistically, we really run a chi-squared. Remember that? Okay, so that could be a possibility. That's not what they're asking here. But I'm looking at this and eyeballing it, meaning looking at the data. These are really far apart from each other. This 309 is really far apart than 862, so we're going to make the assumption that they're not assorting independently. If they were, based upon the number of samples here, it looks to me as if there is some linkage, that there are less recombinants here because there are more parental phenotypes than expected, because these genes are linked. They're close together. The genes for body color are located close to each other in the same chromosome. That's the winner, and that's another way of saying linked genes. Why are they linked? Because the parental phenotypes are bigger than expected, and the expected recombinants are lower, okay, because of these genes wanting to stay together. So we're not going to get new recombinations unless there is crossing over. So that's the winner. Going through the other choices, the traits for body color show complete dominance over the traits of wing shape. This makes no sense. How can you show a dominance from one allele to another? Remember, these are different alleles. The traits of body color is made from a different gene altogether than wing shape. So one gene is not dominant than another. You can talk about regulatory genes turning on other genes, but you can't look at body color and wing shape being dominant. These are different genes, different alleles altogether. So C makes no sense. D, the observed variation in body color are detectable in males but not females. Well, if that's true, then how do we know the phenotypes for females? Okay, if you can't detect them in females, how do we know that the wild type is tan body, wild type wings? That doesn't make any sense. We can clearly see that. All right, so if females are not detectable, then how do we know their phenotype? And therefore, we couldn't do any of this. So the last question makes no sense. Okay. So B is the answer, and it brings up some things that we haven't seen before, and the idea that linked genes are on what? On the same chromosome and close together. Let's bring back a couple of things here that we should be aware of. First and foremost, okay, in meiosis, what we're going to have is a tetrad, which is we're going to have sister chromatins. Now, sister chromatins are basically chromosomes that are the same. They were duplicated in the S phase, okay? And they are going to be close together next to, these are called, the whole thing here is called a tetrad, right? The whole thing is a tetrad. So this is sister chromatins. This could be from the mother. And these are also sister chromatins that come from, let's say, the father. Now, because they're the same type of chromosome, all right, this could be chromosome number 21, let's say, of the human, and this could be 21 too. All right, they're called homologous chromosomes. So they're homologous, means they're the same chromosome with the same genes in the same area, just different versions of the same gene. One could be the broken form if it's a recessive, one could be not. Now, what we do know is that crossing over can occur here. So if we redraw this, okay, if there was no crossing over, the highest percentage of recombinants would be, okay, normally 50%. Now, why 50%? Because you have a 50% chance of taking one chromosome that has the one allele or taking the other when these line up on the middle plate in metaphase, okay, before they separate in anaphase. So there's a 50-50 chance of getting one or the other. But now crossing over adds to the genetic diversity by basically taking one part of one homologous chromosome and basically taking out a chunk of another. So if this is one chromosome, one sister chromatin, this is red will be for the mother, and let's do the blue, and here's the blue, okay. And so this is a centromere in the middle. If we have the two genes, and like them here, if they are close together on the same chromosome, so when we have crossover event, that means that, let's just make a new color here. When we have a crossover event, okay, this will now be the new what part of the chromosome. Both of these will travel together. So the closer they are, okay, the greater chance, okay, of having more parental phenotypes. You start with what you have. So the closer they are, the less chance that you have recombinants. So the closer together the two genes are, the least likely that crossover will occur. Now, if they're farther apart, now I was to blow this up. So if I was to blow this up here, make it bigger, and make this red here, okay, and I can do red. All right, so if on the red, these guys are farther apart, and we'll go back to black. So let's say these two different alleles are farther apart. There's a greater chance of having what? Recombinants to split this up. So you're going to have less phenotypes and more of the recombinants, but you can't be greater than 50% because you can never be greater than just separating the chromosomes. So 50% is the limit. In any case, there's a good little review here about this, and you should be aware of genetic mapping. This is how we basically map chromosomes by doing crosses and seeing the percent of crossover. Okay, and so the percent recombinants. So if I wanted to find the percent recombinants, I would do 309 plus 277. Those are the total number of new phenotypes all over what? The total, which would be 1447 plus 0.1416, and then times it by 100. So all the individuals, these are all the individuals that have a new phenotype, a recombination, okay, due to some crossover event. And then this would be the total, and you times it by 100, and that'll give you a good percentage. You can't be greater than 50 because you can't be greater than the flip of a coin of getting one what? Chromosome then to another. There's no physical way to do that. Any case, remember also in crossover events we're really doing half of a what chromosome in this crossover event here, so you can't increase that value. The bottom line is identify that the Mendelian results are not matching, okay, the observed because of linkage, and a lot of genes are linked, okay. And any case, so 39 is B. Moving forward. Number 40, some cells release active signaling proteins when a membrane-bound precursor proteins are cleaved. That's the key to the entire question. So we've got the phosphobilipid layer, the phosphate head, we've got some kind of probably transmembrane protein, and then we have some type of precursor to a receptor. Now, when these are cleaved by other enzymes, by proteolytic enzymes. So proteolytic enzyme, now this is going to be the extracellular fluid here. We'll do that. And this is the interior or the intracellular inside the cell. So somehow signaling proteins can bind to these receptors only when proteolytic enzymes cleave this precursor. So basically you have these receptors that are not activated yet on the outside. Only when you see these enzymes that are present that will cleave and cut this somehow, and then that form is now will be a receptor that signaling proteins that are released can now attach to. So basically what we have is receptors that are not in their active phase, and we need these types of proteolytic enzymes to kind of activate them, change their shape to get them ready. So the signaling proteins can bind to the receptors once that happens, and therefore activating a what? An intracellular signaling pathway, eliciting a response from the target cell. Okay, we've been down this road again, but what's new about this is the idea that we have these precursors here. It's just kind of a different mechanism to keep, to shut down the receptor. Okay, in any case, this mechanism of activating the receptor binding signaling proteins has been observed in a variety of organisms from bacteria to humans, showing some evolutionary type of descent if we have them and bacteria has them. Many of the enzymes responsible for proteolysis, which is cleaving of that precursor, have been isolated and characterized. Okay? Which of the following questions would be most appropriate to investigate the proteolytic enzymes are evolutionary conserved among the species? Meaning going from bacteria to humans, how do we know that there is some conservation, that what we're using is what the bacteria may have been using? Okay, so let's look at the choices, and this question is all about what choices is the best. Again, most appropriate. How'd you like that? Now we're looking for the most appropriate, okay, choice. So, party people, let's go after it. Okay, are the genes encoding the proteolytic enzymes expressed in the same types in all species? Look at this question, this choice. Are the genes encoding, okay, these enzymes expressed in the same types of cells? They're not even asking, are the genes made up of the same code? They're just saying, are the same types of cells in different species being expressed? Okay, and that's not going to give us a lot of information other than, yeah, they're there, they're being expressed, and there's some, but it's not going to give us the most appropriate way to investigate that we definitely have an evolutionary tie. Yes, it does show that bacteria and let's say humans both have these in similar cells, but it's pretty vague. It's not certainly a home run. If we're looking at the code and compared the code and ran a sequence of nucleotide base pairs to see the genetic code for that, and then compare to see how similar or dissimilar the code is for these proteolytic enzymes. Now we're talking, but just that they're expressed, that just means that, hey, are these types of cells making these types of enzymes? We're not really seeing if in fact these enzymes are similar or not. So that's why I get rid of that choice. Okay, if I look at B, or let's go to C, if the proteolytic enzyme from one species is incubated with a precursor protein from another species, does the correct cleavage? Now remember, that's what this is all about. This is about cleaving, okay, and having the right function to cleave a certain enzyme. So if I take a precursor protein from another species, okay, and I take the proteolytic enzyme from another, will the enzyme cleave from one species, cleave a protein from another to expose the receptor? So what we're really asking is the enzyme that has a particular shape that can actually use this precursor as a substrate in its active site, in its quaternary or tertiary structure, is the active site correct for this? And if it is, then we must have the right R groups that are being attracted to each other from the amino acids. Therefore, we have the right amino acids being put together in the primary transcript of RNA, and therefore we must have a similar genetic code. That's what screams C is yay, yay, yay is the answer. So let's look at choice B. Once the precursor proteins of different species are cleaved, do the active signaling proteins bind? We're not talking about the active signaling proteins. So once the precursor proteins of different are cleaved, okay, do the active signaling proteins bind to the same receptors on different target cells? So it's not about the receptors. It's not about the signaling. It's about, does the protease, remember the question, which of the following would be most appropriate to investigate the proteolytic enzymes? So it's the job of the what? Of the enzyme to actually break apart a certain precursor receptor, not about it's not about the receptor afterwards. If the again, so look at the questions and then look at choice B again. Once the precursor proteins of different species are cleaved, no, we want to look at to see if they can cleave them, because if they can cleave them, they have the shape that's appropriate that matches the precursor in another species. That means this enzyme works for a different species, then this enzyme was made or works with this substrate. And if it works with the substrate, there's definitely a huge evolutionary linkage, meaning the amino acids or the code from one species must be the same as the code for the other to make an enzyme that has the right shape and function for a substrate, in this case the precursor. That's, again, C is so strong. So that's why that part of B here is not true. Okay, now the second part is, do the active signaling proteins bind to the same receptors on different target cells? That's not as important as the job of the enzyme, because we're after what? The proteolytic enzymes doing the cleaving. And D, are the proteolytic enzymes synthesized in the rough endoplasmic reticulum? Of course they are, as all proteins are, because you need what? Ribosomes to put the messenger RNA into proteins, and these will be all alongside the membrane of the endoplasmic reticulum that connects the nucleus, okay, to the Golgi apparatus there, sort of. Any case, so this is a terrible distractor. So in this case, number 40 is C. 41. Transmission of an action potential across a synapse involves the release of neurotransmitters by the presynaptic neuron. This is the neuron before you get to the junction, pre. The arrival of the action potential triggers a rise in calcium concentration in the synaptic terminal. They're telling you that. Okay, and the change in concentration triggers a release of neurotransmitters in the synaptic cleft. So the arrival of the action triggers a rise in calcium concentration in the synaptic terminal. Okay, and the change in concentration triggers a release of neurotransmitters in the synaptic cleft. Which of the following representations of the movement of calcium, sodium, potassium best shows how an action potential is transmitted to the postsynaptic neuron? The post is after. So the question is, okay, who was delivering the signal and which direction? Well, what we do know is in depolarization, okay, we have this spike, we have a graded potential, and then we have sodium ions, rush in to an axon that is already negative. It's negative because the sodium-potassium pump that basically pulls out 3 sodiums and pulls in 2 potassium ions. So we're basically doing what? Removing 3 plus and replacing with 2 plus. So overall, there's going to be less positives inside than outside, and this is going to be negative. That's why it's a negative resting potential. Now, because of that, okay, once there's a stimulus, a graded potential, okay, and the gates are open, sodium wants to rush back in because of a gradient potential. There's more sodium outside than inside, and the inside's negative, so the sodium rushes in. So sodium rushing in has to be part of your choices. Sodium rushing out is not. So that's why I get rid of C and D. So clearly when I look at this, sodium rushing in is telling me that this is an action potential going this way. And potassium rushes out too because, well, for one thing, there's more potassium inside than outside, and when the potassium rushes in, this makes this positive, and K plus is now easier to remove. If we blow up the axon terminal, remember, this is negatively charged because there's less positive on the inside, on the outside. There's less positives on the inside of the axon than the outside. And then we have these gated channels that only open when the voltage starts rising, when there's depolarization. So the sodium wants to rush in because there's more of them. Now there's more potassium inside, so they want to also rush out. And there's gated channels for potassium specific. There's also natural openings. So K does leak out a little bit, but it doesn't leave that much because there's negative charges that because this is slightly negative, the K plus is attracted to it, so it doesn't leave in that much. Eventually it stops. But when the sodium rushes in, when there's a action potential, this makes this positive, and now the K plus, which now is no longer attracted to the negative but has a higher concentration on the inside, leaves, and that's this part coming crashing down is the K plus leaving. So we want the K plus leaving and the sodium going in. So now we're stuck with choice A and B for the same reasoning. Now, when the action potential rushes through, it's going to release neurotransmitters. These neurotransmitters are vesicles of chemicals. They're going to bind to receptors at the end of this junction, and they're going to basically open up the gates, okay, for other ions to rush in. And what ions do we want to rush in? We need positive ions to rush in. Why? To cause an action potential in this area to keep the potential keep going. Remember, this is a resting neuron. It's a post-synaptic neuron, which has a resting potential of negative charges in the middle. So we're going to have to send an impulse. We need positive ions, and that's why A works. Now, if you look at the reading, you see that, what does it say? The release of neurotransmitters, okay, transmission is the release of in the presynaptic neuron, which would be the one before it. Now, of course, it's going to be this one, so I should have drawn it there. The arrival of the action potential triggers a rise in calcium concentration in the terminal. So the deliverer of the action potential increases the calcium by another mechanism, but that calcium now is there in high amounts, calcium plus 2, I should say, okay? And of course, you have a lot more calcium available in the terminal that can now rush in if the gates are opened here. And they're open when the neurotransmitter, okay, is activated by the impulse of the presynaptic neuron, okay, again, gives off these chemicals, these neurotransmitters. Dopamine is an example we talked about, that connect with these receptors, a target, and this is cell-to-cell communication, another example, and that opens up the gates for potassium, in this case calcium, to rush in, which creates another action potential that keeps it going down this neuron. So for those reasons, A is the answer. 41 is A. 42. Number 42. Table below provides a comparison of nitrogenous waste production in selected organisms. We've seen these kind of questions before. Nitrogenous waste is usually excreted in the urine. and it's usually from the breakdown of proteins. So we have nitrogenous waste, different types, solubility in water, and the amount of water required for excretion for different types of organisms. So which of the following statements most consistent with the data in the table above? Another one of those data questions. So look, you have to eliminate choices here. In response to a hypotonic environment. So hypotonic would mean we have a cell who's got a lot of solute particles compared to the extracellular fluid. So clearly, water in a hypotonic environment is going to go from low, I should say high concentration of water to low concentration, or from a position of high water potential to low. So it's going to flow in. So let's talk about this. In response to a hypotonic environment, which freshwater fish live, that's true, they excrete ammonia. Okay, freshwater fish excrete ammonia, so good. In concentrated urine. But wait a minute, the amount of water required for the excretion of their nitrogenous waste product is high. So if they've got a lot of water to excrete their urine, which is their nitrogenous waste, it will not be concentrated. Concentrated means you have a small amount of water for the amount of substance you're trying to dissolve. So even though the first part is correct, the second part makes the entire choice wrong. Let's go to C. Birds secrete ammonia. Well, birds secrete uric acid, so that's wrong. So that's just a reading question. D. The similar regulation of extracellular fluid volume and composition in all organisms suggest a conservation of kidney structure. Boy, this sounds like this could be a good answer depending upon all the words they use. Extracellular volume, composition. People love to choose this, but this is wrong. Is there similar regulation? Look at the different animals. There's what? Different types of compounds. Then there's different amounts of water needed to dissolve them. Okay, so the amount of regulation of water with the different compounds, okay, shows that these different groups, and there's 3 of them, okay, do not have similar regulation, okay, between these 3. So remember they said in all the organisms. If you just picked one, you could probably go there. So there's not a conservation of kidney structure throughout evolution because there's different types of regulations. So, freshwater fish have to give off a lot more water because they absorb more water because they're in a hypotonic environment. Okay, some mammals, okay, are in the water, so it's medium. And then look at the rest of these guys: birds, reptiles, and terrestrial conserve the water. So the type of osmoregulation, that's what we'd use here, is different for the different needs of these organisms in their different environments. So D is not the answer, and so therefore by process of elimination, B is, let's see if it makes sense, the kidneys of reptiles and birds are highly efficient because little water is needed to excrete. Efficiency is the idea that you don't lose something vital, okay? And so for animals like these guys who do not live in water environments, they're land-based animals, they need to conserve the water lost to get rid of their nitrogenous wastes, and they have very low amount of water that's excreted to get rid of their nitrogenous waste. So he's called that efficient because they're able to conserve more of the water that they need because they don't live in a water environment. So it's efficient in the sense that they conserve their water. Think about efficiency and conservation. It goes hand in hand, especially in environmental cases. Okay, so 42 is B. 43. So this is another cluster question, and these ones really challenge a lot of students. So let's get into it. We have a presynaptic neuron and a postsynaptic neuron. We have a what we know as neurotransmitters are going to be released in vesicles from when the action potential travels to the end of the axon, and these little vesicles will eventually attach to receptors that open gated channels for like calcium to come in and to lower the resting. Remember, right now there's a negative resting potential, so positive ions come rushing in. But what this is about researchers investigating a regulation of that neurotransmitter release from the presynaptic neurons, and say that CDK5, okay, is a protein expressed, which means it's made in the axon terminals in this area here, which actually inhibits the movement of the synaptic vesicles to the presynaptic membrane. So what we're saying is these neurotransmitters, which are made at the end, okay, of the axon terminal, or made in vesicles in Golgi, finally put in vesicles by the Golgi apparatus, and they actually are attached to little microtubules, little proteins, and those proteins push them with motor proteins to the surface to be released when there's an action potential. So the model is what prevents these vesicles from just going there anyway is something called a CDK. Somehow a CDK, okay, inhibits these guys from just going. Understand, we want the nervous impulse, okay, to travel through this gap here, this synaptic cleft, but we want to be able to control when these vesicles get to the surface and release the neurotransmitters. We need to control that. We can't always have them have the ability to release them, because if you did that, the neuron would always be on an on position, constantly stimulating. This has to only go on when we need it to make a signal. So there has to be a control mechanism by which we control these neurotransmitters from getting to this part of the membrane and being released. We only want them occurred when there's an impulse, a graded potential and an action potential that travels down through the axon. So that's what they're talking about. So to test the model, what they're going to do really quickly, if you read this, is that we're going to have that green fluorescent protein be genetically engineered, okay, into a tissue sample of nerves. And what they tell you, which is by far the most important part, is that in acidic conditions, which we know is acid when the pH is low, okay, you have to know acidic conditions mean the pH is low. Okay, the GFP protein has no color. But in alkaline conditions, which is basic, okay, we know that the GFP is a higher pH. And they said there is a bright green fluorescence when we have a higher pH. So if you go back into the model here, notice the synaptic cleft is a higher than 7 pH. So this is basic. So we can tell when a neuron fires, as long as we genetically engineer some of the cells in the axon to make the glowing fluorescent protein to be released along with other vesicles. And when they get into this space here that's a little bit basic, they'll glow green. So number one, based on the model, which of the following best explains why a bright green fluorescent was observed following a stimulation of a presynaptic neuron? Well, the only way that can happen is if the fluorescent green protein was made in the same area that the neurotransmitter, they're in vesicles, and they're not blocked, okay? Somehow the model's working so they're not blocked, and they're released into a space. Notice the cytosol, which is the interior of the nerve cell, is a little bit acidic, and we just told you, or at least the information said, that when the pH is low, there's no color, so we don't see it until these vesicles get to the surface and are released in the space here in a basic range, and that's when they become colored. So you needed to know that to answer this question. I know we've said before that you should be able to grab the easier questions in the beginning. In this case, it doesn't work true to form. So GFP synthesis was activated when a gene taken up by the neuron was switched on. It's not about gene expression. It can't work that way. We have to be a faster method. Okay, it can't be that when we get an action potential, we have to make a protein. That would just take a little too long. GFP was stored in the cytoplasm of the neuron, okay, and was packaged into synaptic vesicles. Okay, but how does that explains why there was a bright green fluorescent only when there was stimulation or an action potential? Okay, so this was probably done no matter if there's an action potential or not. GFP contained in synaptic vessel moved into the synaptic cleft by exocytosis. Yes, because of the stimulation of the action potential, okay, the vesicles weren't inhibited by CDK5, so therefore they got to the end of the axon, released into the synaptic cleft that we just said was what? Basic or alkaline, and they glowed. Exocytosis just means that the vesicle, once it got to the membrane, kind of became part of the membrane and just released it outward. Endocytosis is when the membrane kind of engulfs it to make a vesicle. So C is the best answer. There was no way around. You had to dive into all of this to understand that. And D, the bad choice, GFP present in the synaptic cleft was reabsorbed by endocytosis, which is the opposite. Well, that's true. It probably is. But if it's being absorbed by endocytosis, we know that the inside of the nerve cell is what? Is acidic. So you wouldn't see a color by the absorption of the green fluorescent protein. You would see no color. So that's the change that doesn't make any sense. They said explain when you do have a color. So D is not the answer. So 43 is C. 44. So number 44. Which of the following best explains why GFP, the green fluorescent protein, might exhibit a bright green fluorescence in an alkaline, which we notice is the base conditions, but not in the acidic conditions? So why is a protein, right, able to undergo fluorescence, which with some type of energy it glows, okay, in a certain base solution but not acidic, which means in acidic conditions, okay, it doesn't have the ability. What do we know about proteins? Proteins are affected by their form and it affects their function. This is an important piece to understand. So something in the acidic conditions probably messes around with the amino acid building blocks. Remember, these are polypeptide bonds, and the shape of these proteins depends upon how the R groups from the amino acid of one part of the chain attracts the R groups of another. So how these amino acid chains fold on each other to create the three-dimensional shape creates their form, and their shape, depending upon their shape, creates what kind of function they have. So somehow the acidic condition breaks up some of these R groups in certain ways that the actual shape changes, therefore its form changes or its function changes in acidic conditions. So which choice is speaking to form and function? Let's look at A. Addition of H plus to the GFE at acidic pH changes the shape of the protein. So why are they talking about H plus? Well, we should have learned, and we have learned, that acids are acidic because they increase the H plus ion concentration. In fact, pH is the positive value of the H plus ion concentration. If you remember, 10 to the negative 8 for an H plus ion concentration, the P positive value of the exponent is equal to 8. So this number would be a concentration, a molarity. So that's why A is by far the best answer. The addition of H pluses, which come from acids, to the protein at an acidic pH changes the shape, and therefore when you change the shape, you change its function, and therefore it loses its ability to fluorescence. That's the home run. The rest of these choices are just silly type of confusing choices or distractors, I should say, that have nothing to do with form and function. So 44 was a form function question. All right, so number 45. Before I get to 45, though, one thing I should say is that when you change the form by the acidic conditions, we would call that denaturing. Just another word to do, denaturing. Okay, that's changing the form either by heating or, in this case, adding an acid. Okay, here comes number 45. So number 45, I call this a straight reading question. Which of the following observations best supports, here we go with the best choice, the hypothesis that CDK5 negatively regulates neurotransmitter release? This means that this chemical stops extra vesicles from reaching the end of the axon to release the neurotransmitters that will do what in the synaptic cleft? Travel through it to receptors that will continue the propagation of the signal through the postsynaptic axon. So the entire idea of the entire grouping question, or cluster question I like to call, is due to, in fact, that the CDK regulates these vesicles somehow. Somehow having CDK, okay, 5 in this case, regulates how these go. And we're saying negatively regulates, meaning it stops it. Okay, that's what this model's about. So which of the following choices, we're going to have to go through them. Number A says introducing of the CDK5 protein into neurons results in the movement of synaptic vesicles into the plasma membrane in the absence of any stimulus. What does that mean? Well, that would mean that we're going to have neurotransmitters going from the end of the axon signaling this axon to open its channels so like calcium plus can rush in to continue the signal. We're going to have the signal continuously on? No, we want to control the on and off of these signaling. So that doesn't make any sense. You're saying introduction will infer basically the introduction of the neurons results in the movements. They just said negatively regulates, meaning CDK5 should not increase the movement, it should decrease the movement. So this is going the opposite of a negatively regulation, or negative regulation, or negative feedback, if you wanted to say. It's not negative feedback, but the concept of this stops the vesicles somehow from getting to the plasma membrane to be released to keep the axon, what we call, in an off position until there is a stimulus. Okay, uptake of a gene encoding CDK by neurons result in the movement of the vesicles to the plasma membrane. Well, that again is going exactly against what we just talked about. How, if you have this should what? This should prevent the vesicles from getting there, the neurotransmitters. So they're saying it results in the movement of the vesicles when it should be stopping the vesicles. This is the opposite of what the statement said. It's a reading question. Suppression of the negative compound that should stop it, okay, in neurons inhibits the movement. Wait a minute, if you suppress the negative regulation, then this should be able to move. So if you suppress the negative regulation, you should have an increase in the movement of synaptic vesicles dumping the neurotransmitters, not an inhibit. Again, another choice going the opposite of what the concept is about, the idea that this chemical somehow prevents, or this protein somehow prevents the vesicles from getting to the plasma membrane and releasing the neurotransmitters, keeping the axon in the off position. So by process of elimination, D must be the answer. Inhibition of the CDK activity, so stopping CDK, results in the increased movement of synaptic vesicles. That's the answer. If you remove the negative regulation, then therefore the vesicles will get there. Okay, and look what it says, to a plasma membrane in the response to a specific stimulus. So a stimulus somehow, okay, inhibits CDK from stopping these vesicles. All right, so that was, to me, a reading comprehension problem there. And so 45 is D. It's the only choice that talks about CDK in a light that gives it a negative regulation. All right, and so if you lower CDK5, these guys can get to the synapse membrane better when there's a stimulus. So the stimulus somehow controls CDK, and that's how we keep the axon in the off position. All right, so number 45 was D. Number 46. Number 46, based on the model, and the model, of course, that we're looking at in this problem is CDK5 is a molecule that inhibits the vesicles that have neurotransmitters from getting to the plasma membrane, okay, when an axon is stimulated, so that the action potential travels through the axon through a synaptic cleft to the next axon. So which would most likely mechanism? So now we're going to basically use our understanding of biology here to make some sense of this. Okay, so let's go through our choices. A says CDK5 adds methyl groups to DNA. Now this is called methylation. This is called, we add methyl groups to DNA. What this does is it adds methyl groups to certain nucleotides that pretty much prevent the nucleotide from attracting others. So if it's in the promoter region of the operon, what it does is it represses the gene so that you can't have something attached to it to increase the rate of transcription. So methyl grouping or methyl groups adding to DNA can repress or inactivate that part of the gene. It's one way that we can control for the gene expression. All right, so adding methyl groups altering the expression of genes required for synthesis of neurotransmitter. The way CDK5 works cannot be by regulating or altering how genes are expressed, because neurotransmitters have to be quickly shuttled to the surface and released to quickly have this action potential that fires down a nerve very fast to go from one nerve to the other. If we have an action potential, and then in order to make the other nerve fire as quickly without a lot of lag time, if we have to express genes, that would take too long. So the type of neural control, or the type of control that CDK5 to controls how these vesicles get to the end, cannot be based upon the expression of the gene. It's got to be about how these vesicles physically get to the plasma membrane, okay, and releasing by endocytosis. So that can't be it. B, CDK5 promotes the rearrangement of the lipid molecules of two bilayers. I'm not sure what that does and how that works, and if in fact that is true, but you're talking about rearranging the lipid layer. We need to talk about what? Getting the vesicles, okay, to the position so that they can be released, and quickly if there is what? If there is a what? A stimulus, okay, an action potential. Now CDK that's present stops it, so somehow the stimulus, okay, is inactivating the CDK5 so that these can get there quickly. So again, if you get lost like I am, reread the question. Based on the model that CDK5 is a preventer, a negative regulatory or negative chemical that prevents the vesicles from moving, okay, based on this model, which of the following describes the most likely mechanism how it regulates? Well, we can't do the methyl groups, so we go to B, promotes the rearrangement of bilayers. No, it has to be something physical of helping, getting, or stopping the vesicles from moving from one place to the other. CDK5 alters the activity of other proteins involved in the movement of synaptic vessels. That's the ding, ding, ding home run. Now, as a reminder, vesicles just don't move arbitrarily from, let's say, the Golgi apparatus to the cell membrane. They don't. Okay, they move in a distinct way. Normally they travel through microtubules, and there's motor proteins that help move the vesicle from one place to the other. So there's an arrangement of proteins, protein fibers called microtubules, where vesicles will move along with motor proteins to pull this vesicle in a distinct way, kind of like you know, train cars going to a certain spot, you know, railroads. And so the structure of the atom is really impressive that way. So 46 is C. Let's finish this out. CDK5 binds to gated channels in the postsynaptic memory, resulting in diffusion of calcium ions. Well, we know calcium ions will go through these gated channels once the neurotransmitters hit a receptor to open up those channels, or there's other mechanisms, but somehow these receptors for the neurotransmitters that are eventually released, if this presynaptic membrane has a action potential, will send out these vesicles, and that they'll hit a receptor that eventually allows calcium ions to rush in. Remember, this is also an axon here that has a resting potential, and so the positives rush in and you have a continued action potential. So that seems like it could be there, but we're not talking about the post synaptic membrane. We're talking about the presynaptic membrane, the vesicles moving to the end of the axon, okay, in the presynaptic membrane. So that's why D doesn't make any sense, and again, C is the best answer, okay? So 47. Number 47. Previous experiments indicate that CDK5 is active only when attached to a protein called P35. Don't forget, the entire model of this problem is that CDK5, that protein, somehow inhibits, stops vesicles carrying neurotransmitters from getting to the plasma membrane to be released to cause a what? Action potential to the next neuron in this synaptic cleft. So if the CD5K is regulated negatively by CD5K, easy for me to say anything today, it seems like, that it says here is only active to stop the vesicles when the protein P35 is attached. So which of the following best predicts how P35 might play a role in regulating neuron function? All right, let's go through this. Elevated intracellular, inside the cell, levels of P35 result in increased synaptic activity. Well, wait a minute. CDK5 stops the vesicles and is active and can do so. That's what it means to be active when P35 is around. So if you have elevated, which means greater amount of P35, that means that that's going to make sure that this is working properly. It activates it so it can stop. So therefore the vesicles do not, with the neurotransmitters, get to the end of the axon. So the activity of the synaptic will decrease. You'll have less neurotransmitters. So therefore the signal will go down this axon but stop here. Degradation of P35 results in increased synaptic activity. Yes, if you break down P35, that no longer can bind to this CD5K. So if you break this down, what happens is now this is the inactive form of CD5K, and now the vesicles can get there, and you're going to have neurotransmitters flowing to the receptors to open up gated channels for calcium to rush in and cause an action potential. So that is the best answer. Going through your choices, if you're not sure, reabsorption of P35 from the synaptic cleft. Well, if you take in P35, what's it going to do? It's going to bind with the CD5K and probably cause a conformational change of the protein so that now it's in the active state. And once it's active, it stops the vesicles, and it will decrease the synaptic activity. Remember, the synaptic activity is the ability of the neuron to release the neurotransmitters from a stimulus or an excited or propagated signal action potential that travels down this end of the axon, so to speak, if you can think of it that way. So if I limit the vesicles from getting to release the neurotransmitters, I limit the ability of this axon to stimulate the next axon in this little cleft area. So that's why this makes no sense. So if you take in P35, it'll activate this to stop it and should reduce, not increase, and that's why C is a bad choice. D, attachment of P35 to the synaptic vessels results in increased synaptic activities. Makes no sense because, I can't speak, makes no sense because the P35 has to work in concert with the CDK5. This choice is just skipping CDK5 altogether. This makes CD5 active so it can do what it normally does, which stops the vesicles. So that's why 47 is B. Number 48. Number 48, based on the model, and again the model here is that CD5K is a protein that prevents vesicles from getting to the end of the axon to release the neurotransmitters that will open up the gated channels so that calcium can come in and stimulate the next postsynaptic membrane so that the action potential can travel from one axon to the other. So Based on the model, which of the following best explains how regulation of the neurotransmitter release, which is done through CD5K, it's the model, might increase the range of responses to a stimulus. Now we know pretty much there's a stimulus response, and that stimulus is an action potential, okay, where we have a spike in positive charge that flows through the negatively charged axon. So this action potential with this graded response into the spike here travels through at high speeds. But how may we increase the range of responses in a nervous system where you have multiple neurons connected? Well, in the absence of any stimulus, neurons can still release neurotransmitters. That'd be a bad thing. Okay, we do not want neurons to be releasing neurotransmitters to constantly have our nerves firing. We want to make sure we fire for the right reasons and control the rate of that signal. Okay, so A makes no sense. B, different neurons in the same neural network. So I guess that's basically a lot of, if we've got some kind of network of neurons, that's basically, let's say in the brain where you have integration, these different neurons can release different amounts of neurotransmitter, and that may be a way for us to make the signal more complex. Think about it, our brain is made of nerves, a bundle of nerves connected to nerves. How do we integrate nerves into thoughts and numbers and everything else that happens? And so one way to increase the complexity would be to increase the different amounts of neurotransmitter in that region. And that could be controlled simply by this CD5K model, okay, that we've been talking about, that can control how much. So that seems to be a great answer. Let's go through our choices just to make sure. In the depolarization phase, that would be right here. That's that spike through the sodium ions coming in through the channels, gated channels. Postsynaptic neurons can adjust the amount of neurotransmitter bound. Wait a minute. Postsynaptic neurons are these neurons that the flow of the action potential flows down one into this synaptic cleft of another neuron, and it'll travel this way. So we're talking about neurotransmitters bound to the receptors. The postsynaptic, this is the pre, okay, PRE, right here, and this is the post. But wait a minute, neurotransmitters come from the presynaptic. Pre is before. So that would not be a great example. In the depolarization phase, where sodium ions or calcium ions, if it's from a new, but we're talking about the pre, so that this would be sodium ions, are rushing in and creating this action potential. Okay, postsynaptic neurons can adjust the amount of, not really, they just have what? They just have receptors here. So that doesn't make any sense. The amount of neurotransmitter of vesicles that are getting to the end, like here's a vesicle, getting to the plasma membrane, as I have drawn for other questions, is dependent upon the regulation of CD5K, but that's nothing to do with the postsynaptic axons here, okay? Controlling for how much neurotransmitters that are bounded would be controlling the amount of what neurotransmitter released. So that's why C makes no sense. You really want to control the neurotransmitters, and where are they released? They're released in the presynaptic, before you get to the synaptic cleft here, this gap in between. It's due to this ion, okay, this ion, or this axon I should say, right there. It's due to this one right here, and not this one. This is secondary. If we look at D, the recovery phase, that's going down. That's because the potassium's leaving, and those channels eventually close, and you get that undershoot here. In the recovery phase following a stimulus, enzymes can be mobilized. That means you get them together or made to degrade the molecules present in the synaptic vessels. Well, that's important because if you release neurotransmitters, they're going to connect to these receptors and then open up gates. We want some enzymes to quickly degrade these so that these signal molecules, these neurotransmitters, are not in the cleft anymore quickly to turn off this signal. Okay, and that's true, but it doesn't answer the question. And if you get lost in the sauce, what is the question? Based on the model, Which of the following best explains how regulation of the neurotransmitter release might increase the range of responses? The only different types of range of responses means that possibly you can give off enough neurotransmitters to hit the receptors maybe in this cleft, but maybe they can move to another synaptic cleft. And that's controlled by how much vesicles are able to produce and release that signal, that neurotransmitter. So that's why you're able to control maybe your response, but maybe one axon can excite three junctions or synaptic clefts if they produce a lot more, okay, of the vesicles that contain a neurotransmitter. So that's why B is the answer. 48 is B. Number 49. Okay, number 49. In a species of cactus, the number of spines on a plant is genetically determined. Okay, the graph above shows frequency distributions for populations of cactus species growing in the presence or absence of two herbivores. So we have two types of plant eaters. We have the pecaries, a type of pig, and a wasp larva, okay, the little guys before they become flying guys. Which of the following best accounts for the different frequency distributions of the graph? So if we look at our graph, we can see with both herbivores that this graph is higher and it peaks higher, but the range of spines per plant, those little pricklies, is a lot less. But when you have neither herbivores, the distribution of spines in the populations is a lot less. Excuse me, check that. When we have neither herbivore present, the range of spines per plant is greater. Okay, there's a greater distribution. There are more individuals with greater differences, high and lower. You would say, okay, that the standard deviation is smaller for this line where it's greater. There's a better spread without the herbivores. So what the herbivores are doing is they're providing a negative selection, or they're providing a selective pressure to eliminate, okay, individuals with a low number or a high number, okay, of spines. So we call this stabilizing natural selection. Boy, that was written so bad. So stabilizing selection. And the reason why it's stabilizing is because essentially it's a type of natural selection in which the genetic diversity goes down and the population mean stabilizes on one particular trait. And the trait would be to a certain range of number of spines per plant. Now, why is there a selective pressure to eliminate alleles of individuals who have high or low numbers? Why is there a stabilizing natural selection? Well, party people, the reason for that has to do to the herbivores. So the pigs probably don't like what? The great number of pricklies. So if you were to eat your favorite food and it had a lot of these spines on it, you wouldn't eat it. Okay, so the pig is probably not going to go after these guys. So we probably would expect these guys to live more. The pig is probably going to say, you know what? I'd rather eat the plant with less spines. So this area here, sorry for the crazy drawing graph, but this area here is where we're going to have our stabilizing selection due to the pigs. The pigs are going to eat all of the plants that had the lowest number of spines, which means you're going to naturally select, and apply a pressure for those individuals that have more. But we would expect this to move over, not just become a spike, so there must be another selective pressure. So it must be that the wasp's larva, okay, love to eat the plants that have more. It has to be in order to have this little skinnier, higher curve with more individuals with a range in between that stabilizes. So let's look at our choices. Okay, A, pecaries eat cacti with the greatest number of spines. Well, if they eat why would they do that, by the way? They like to eat the spines. It hurts their face. So that doesn't make sense to me that that would do that. But let's just continue. Let's just pretend that could be. And the wasps show no preference. Well, it can't be true. Okay, even if you think that there's a possibility this might be true, the second part cannot be true. The larvae definitely are showing a preference because we are wiping out both sides. So both herbivores are causing the stabilizing selection by wiping out the extremes, okay, in this case. So we get rid of A. Now looking at B, pecaries eat cacti with smaller number of spines. That makes sense. That I like, and that would limit this part of the curve to make it what? Look like this right here, a little taller. So therefore, that makes sense. And wasp eat cacti with a greater number of spines, and that would put the selective pressure against those individuals with higher. And then we're going to left with a population that only has good fitness for a smaller range of spines somewhere in the middle that stabilizes that. And that's why B is the best answer. Okay, the rest of these, okay, are just talking about nonsense that doesn't stabilize and work off both extremes. Number 50. Scientists are studying several populations of finches on neighboring islands of the South Pacific. Previous genetic analysis has shown that a single gene controls tail feather length and finch populations, and that the allele for the long tail is L. Okay? And the allele for the short tail is little l. It's recessive. On two separate islands, the scientists recorded the number of finches with long tail and the number of finches with short tail feathers. Here are the results. Okay. If the two finch populations are each in Hardy-Weinberg equilibrium, now this, if they're in a Hardy-Weinberg equilibrium, that means there's no evolution, which means there's no gene flow, there's no natural selection, there's no mutation, no drift. Okay, there's basically the percentage and the allele frequencies, the frequencies of the phenotypes of people with certain expression of genes stay constant. Okay, and that's important. They're isolated from each other. Which of the following graphs displays the relative genotype frequencies? Now, genotype would be, well, okay, homozygous dominant. All right, and heterozygous dominant. Let's look at the questions here. Yeah, so we have homozygous dominant, heterozygous, and then homozygous recessive. So my gosh, this is a question where if you could look at this data and try to come up with those frequencies, that would be really helpful. Now, what we do have is we do have the individual recessive, and the jumping off point would always be the individuals that are the recessive. So this is little l individuals. The problem with the long tail feather people, that phenotype could either be homozygous recessive or heterozygous, and then or homozygous dominant, heterozygous or homozygous dominant. So we're going to have to calculate here to see where we fall in. Hard to try to do this by looking at it. So this is going to require a calculator, and hopefully we can do the minimum amount of calculations to estimate the rest of these and cancel off the bad choices. So we're going to have to do some math here. And so what we do know is that we have all the individuals that have short tail feathers. And the individuals with short tail feathers, okay, these are people that we would call Q squared. These are the homozygous recessive. And what we have is, we have 598, okay? Now, to get the frequency of the individuals that are Q squared, which equal homozygous recessive, okay, to give that short tail feather phenotype, we're going to have to find the frequency, which is going to be part over total. So we're going to find all the individuals that carry both of these alleles, okay, and we have them there. That's what the Q squared is. And then we're going to divide them by the total, which is 2180. And we're going to come up with a number, and that's the frequency. So how many people have it over the total individuals? So 598. 598 divide by 2180, and what we get is 0.27 approximately. So we know that 0.27 is the frequency of the short tail. So basically, I mean, I'm not going to do every one of these. So let's go see if the island 1 people, okay, will have that. And what we know is the island 1 is the lighter shade. So 0.27 for the island 1. Well, looking carefully, 0.27 for island 1, then that's 0.27 should be around here. So I'm going to get rid of that choice right away. This choice is less than 0.1. Okay, so I get rid of choice A. Then look at C, 0.27. Boy, that's about approximately right for that one. And this one's way, way below 0.1. So just by doing one math problem, I'm able to estimate that C is my answer because the recessive individuals, their frequency in the population, okay, for the island 1 is 2.7, and there's no other ones that are close to that, so C has to be the answer. So there was one little math question I had to do, nice and interesting. I haven't seen that before, but certainly hopefully a very easy one. The key to this is understanding that these were the individuals, and anytime we deal with individuals, we either have P squared or 2PQ, or we have what? Q squared. The easiest, the jumping off part, I can't speak still, are the individuals that are Q squared. We can't use long tail feathers to come up with the individuals because they could be the homozygous individuals, or heterozygous individuals, or the homozygous dominant individuals. Okay, so our jumping off point was finding that. And I just did one and see which one was closest. So therefore, the answer is C. Number 51, based on the universal genetic code, I would guess DNA, which of the following represents the correct polypeptide that will result from the translation of the messenger RNA molecule shown below? So what we got is we've got messenger RNA, and it was, of course, transcribed from DNA. So we have the basic transcript here, the primary transcript of the messenger RNA. It could be capped or not, but we're going to start with beginning with the start codon, and we're going to basically figure out the polypeptide. So when we go here to the triplet codon above here, let's go find the start codon. And when we find the start codon, what we do is we see that it's AUG. And let's go find that guy. And when we do, we see AUG right here. Now, it's the start codon, but it's also going to code for methionine, so MET is definitely coded for it in the beginning. Okay, then we have GUG, so we go GUG, and when we find GUG, we see that's Val. ACG, so we have ACG, and THR is the shorthand abbreviation for the particular amino acid. I'm going to stop there because we can just eliminate our choices. Notice we start with the methionine always, so we can eliminate A and D right away. And then we have Val next and THR next, and we're done because the other choices don't have those three, so C is the answer. There's no more to this, people. Choice C. Number 52. The data above represents the results of 3 different crosses involving the inheritance of a gene that determines whether a certain gene organism is blue or white. Which of the following best explains the mechanism of inheritance? So let's just dive in here and see what this means, because I can't answer any of these questions if I don't get a good feel for this. So in the first cross, I have a blue male and a blue female. Okay, I do not know anything about who's dominant or not. What jumps out at me, though, is that there is a 0 amount of phenotypes. There's 0 white females. Also that jumps out, there are 0 blue males and 0 white females. So I'm having scenarios, okay, where the certain female is not showing a certain phenotype in the cross, and certain blue males in a certain cross are having no phenotypes. This is screaming, okay, a sex sex-linked gene. Now, a sex-linked gene means that the allele is carried on one of the sex chromosomes. And most of the time, okay, it's going to be the X that the alleles are carried on. There are some alleles that are carried on the Y, but not many, and most of those are male-based genes. If we remember our work this year, an X chromosome's pretty big, but a Y chromosome's tiny. So there really isn't anything other, the genes on the Y chromosome tend to be just male-dominated genes. So because this chromosome is so big and you have the additional amount of space for other types of genes besides the female characteristic genes, this is going to be probably an X-linked, okay, recessive disorder or recessive type of inheritance pattern. Now I say recessive because when I look at the female, there were no females that were white. Now females are X and X. And if there are none that are white, that means for me, I'm thinking that there's a recessive allele. Let's just call it uh, just a little insignia there. These are the recessive alleles. You need to have both of them there. It's harder for females to express a recessive X-linked phenotype because both X's have to be the broken gene or the recessive allele. So that's why I'm seeing 0 white here. Now, besides that, what kind of cross would give me that? And so I'm going to have to get some space here to think about these crosses. For me, I'm going to have to do a cross to understand this. It looks to me, though, real quickly, that this is going to be X-linked or recessive type of scenario. So let's just find some space here and see if I can make some sense of these crosses so that I can go and attack this. Now, if you looked at choices A and B, they're talking about autosomal dominant. Now, that autosomal just means non-sex linked. But because the number of phenotypes are so different in some of the males and females, man, I'm telling you, it looks to me like that cannot be the case. So to me, autosomal dominant is a non-sex linked characteristic, which means the sex is going to have no effect, but apparently there is an effect. So it's between C and D, but I need to do some Punnett squares here to make some sense of this. So think about this. I'm not going to have any females that have the recessive allele. And because of that, when I mate a female XX with a male, okay, just taking the hypothesis that the white is the recessive allele, okay, so if we think about the blue male, the blue would be a dominant. Now, because I'm going to say it's being carried on the X, because it normally is, because the X, of course, has a lot more room, I'm going to put a big W here. So to be a blue male, as far as I'm concerned, you'd have to have the dominant allele on the X. And because you don't have a Y, whatever you have on the X, boom, it's what you have. Now, we're doing this with a blue female. Now, a blue female could have two what? Could have two homozygous dominants here. Okay. Now, because I have mostly, I do have some white males, okay, and I'm saying that the white is the recessive allele. I'm going to, the only way for to have that is that one of the X chromosomes have to have this broken gene or recessive allele. Has to be, because how do I get offspring that are white if I want to say it's the recessive? Somebody has to deliver that. So when we do the cross, okay, what do we have is we have an X big W and an X big W. So this is going to be a blue, these are dominant alleles, blue female. Okay, and we had plenty of blue females. And we do it again, we have XW, big W, and then we're going to have the X with the recessive allele. So because the recessive allele is going to be masked by the fact that there's still another chromosome that carries the dominant trait. So both of these, more than 50% of your offspring in this cross will be blue. And look at that, 124, it looks like that's about half of these two added together. Now let's do the males. X and Y. Well, we have an X that's dominant, and then we have a Y that's not. Well, the Y doesn't have anything on it because it doesn't carry that allele on that chromosome. So because there's only one X that's dominant, this is going to be a blue male. And we have blue males. And then, of course, we're going to have the X that is the recessive with the Y. And because there's no other X to mask it, this becomes a white male. And notice that's going to be a quarter of, there's going to be a blue male and a white male. And look at that. They're about almost equal, a quarter of the total. Okay, so this is kind of confirming the fact that this is a sex-linked gene. And let's see here. The allele for the white is an X-linked dominant. No, it's an X-linked recessive for this to work. And the reason why it works this way, because it's recessive, just having the one allele will give you the white for the male. So the males are a lot easier to become white. Okay, so that for that reason, I can get rid of that, and then you're left, because A and B is talking about autosomal dominant. This has nothing to do with sex-linked, and saying it's not sex-linked. Then D has to be your answer. But the D is saying the allele for blue is an X-linked dominant allele because there are no blue male offspring in cross 2. Okay, so what does that mean? Now I know I said the inheritance pattern is like a what? A recessive X-linked gene. But they're talking about blue now. For blue males, okay, we just talked about only having one X. You'd have to have an X that's dominant. So it's an X-linked dominant allele for the blues. Now, why does it say that because there's no blue male offspring in cross 2? Let's try to explain that. You don't have to. By the process of elimination, D is your best choice, eliminating A and B because those are not X-linked type of scenarios. So we put these in. We have our X. Now let's talk about this. We have our white female. In this case, you're going to have two X's that have the broken gene right there. It's the recessive allele. And then we have the blue male. The blue is an X and a Y, but this X, of course, is going to be the dominant, okay, white. And when you do this, you have the X broken gene, and you have the X with the white. That's the female. And if you notice something, broken gene here and the X with the W, both of these females, or half of them, are going to be blue, the dominant. If you notice that the biggest amount of all the females are blue, and there's 0 white because you don't have another an X from the mom and X from the dad to make a homozygous recessive. You can't have it. So that's kind of explaining things. And let's explain the male. We have an X broken Y, so that's going to be what? A white male, and then you're going to have the Y again. Dad gives the sex, right? And then the same thing. So you're going to have all boys, all the males are going to have the broken X allele, and therefore they're all going to be white, and there's 0 blue. So the question says the allele for blue is an X-linked dominant allele. That's right. To get blue, it's a dominant allele on the X-link. Okay, sounds a little weird, but that's how it works out. You don't have time to millions of crosses, but you should have been able to figure out this was X-linked, and then figure out, you know, from maybe just one of these that C wasn't the answer. You do one more to verify what we're talking about, but 52 is D. Okay, so number 53, the final multiple choice question. Okay, so a table shows the changes in the allele frequencies of a specific gene in 2 populations of randomly mated small mammals. After 30 years, the populations inhabit adjacent equatorial islands that have similar topography and climate. Which of the following is the most reliable conclusion that can be drawn from the analysis above? So what we see here in 1980, we have basically the frequency of this allele, which is dominant, and then we have the frequency of the recessive. Notice 0.7 times 0.3 gives you the 1. So 30% of all the alleles at a certain locus of a chromosome is dominant, and 70% is the recessive. And in 2010, you have 0%. You have the death of an allele, and you have what? 100% R. And in population 2, it's 37% R, 75% R, 30 years later, and so forth. The changes here. So what we're seeing here is evolution. Evolution because there is a change in the frequency of alleles. Okay. Now, evolution isn't just a natural selection, it also changes in the allele frequencies, and there's many ways that that could happen. Now, which of the following is the most reliable conclusion that can be drawn from the analysis above? For me, when I see the death of an allele just go like that reminds me of the activity that we put in the Excel spreadsheets when we watch the frequencies drop with small populations. And if you've got a small population, since they're only on um, uh, an island, what can happen is genetic drift. And that jumps out to me as the answer. Genetic drift are chance events. Just by chance alone and only have a small population, you can actually lose some alleles. Now, how could that happen? Well, it's kind of like rolling or in this case, flipping a coin. This is the head of somebody maybe. Okay, I'm not sure where, but the point is that's the heads and the bottom is the tails, and you flip a coin, there's always a 50% chance of getting heads or tails. But, party people, the problem here is that if you flip the coin only a few times and you have a few number of alleles, there's a great chance that you could have heads, okay, 4 times in a row, just like having 4 boys in a family. Even though there's a 50% chance to have girls and boys, there's definitely a chance event where you can have all boys. Now, certainly, you could see that to be a problem if you've got a really small population and just by chance all of the male and female birds who, let's say, moved to an empty island, they all were boys, then that means you would lose the female allele just by chance. Okay, so there's an example of an extreme example of genetic drift. So genetic drift are basically the drift of alleles based on chance events because of small populations. Now, of course, if we were to flip this coin 1,000 times, okay, approximately 500 flips would be what? Tails, and 500 approximately would be heads. Okay, but with small number of flips, there's a great possibility that you can have no tails and all heads or likewise. And that's what happened here. Okay, over time, we lost this allele. They died out just by the chance events of getting one chromosome that had the allele and not getting the other chromosome when they segregated from each other. And so we lost the dominant allele or the chromosome that carried the dominant trait. Let's put that R there. Okay, just by chance of more gametes getting this one. Now, it's not just the gametes getting it. Maybe that's the gamete that actually got fertilized. Remember, we make gametes, we're going to make gametes of both, but this by chance is the one that was successful in fertilization. Okay, or even if this one was successful in fertilization, maybe it didn't have any children. Again, these are all chance events. So A is clearly the winner here. Okay, if we look at the choices, if we don't see that right away, sometimes looking at the choices help us understand that, okay, there's only one correct choice and we can do the process of elimination. So B, population 2 is in Hardy-Weinberg equilibrium. No, it's not. Hardy-Weinberg equilibrium means you have no evolution. Here you have a changing of the frequencies. Hardy-Weinberg equilibrium states that the frequencies would stay constant. Why would they stay constant? Because there's no mutation, there's no natural selection, there's no preferred mating methods. The mating is random, so there's no genetic drift of alleles from other populations, and the population size is large so that there is no genetic drift. So these changes that we see here are showing that it's not at equilibrium. Equilibrium means, as we always understand, is boring. Things stay the same. So B can be canceled. C, selection for R is occurring in both population. Well, if there's a selection, a selective factor that makes the allele R more advantageous, okay, then we would see in population 2 an increase by natural selection over time of the R, but we're seeing a decrease over time. So if there's a selective advantage or a reproductive advantage of having R, then we would see that evolution, but we're seeing the opposite. So that's why I'd cross off C. And the reduced frequency of the allele R in population 1 will eventually lead to extinction. Um, Not necessarily, but what it does do is it definitely lowers the genetic diversity, and that could lead to extinction. But remember, this is only one what? Allele on one locus of a chromosome. So just because we lost one phenotype or allele doesn't mean that all is doomed, because you have so many other alleles. You have thousands, 30,000s for humans, different genes that you have. So it certainly lowers your diversity and makes you more susceptible to not have an allele that will match a changing environment in the future. But then remember, this is only just one. This is the evolution of one allele or one gene. So it's not doomsday by any means if that actually happens, that you lose, let's say, one allele or one express trait. So D doesn't make any sense in the sense that this is only talking about one allele and you have so many more. So A is definitely the best answer. And that concludes the multiple choice. And we're going to go on to now the grid-in math questions, even though we've seen some math questions in the multiple choice for the first time. Continuing. Number 121. The cladogram above shows proposed phylogenetic relationship for several vertebrates. Several derived characters, these are characters that made them branch off and diverge, are indicated by the cladogram by the numbered labels. Based on the information presented, which of the following derived character is shared by alligators and manatees? Okay, well, let's go find alligators. Here's an alligator, and here's a manatee. Now, what are they shared? Well, they must be in the same clade, and the clade is a fork. And so if we look at the scenario here, all these guys are in the same fork, and all of these species, lizard, alligator, hawk, and manatee, and the elephant, are all similar because they branched off somewhere because of the amniotic egg that they have. Okay, so this egg, or amniotic egg, branched here, and all of these species have that characteristic. That was what made them differ by the others, the salamander. So the alligator and the manatee have number 3 factor, or number 3 derived character, that this clade, this big fork, they all fit in. So just 3 is what you needed to bubble in. So where you see the number 3, 1, 2, 3, you'd bubble that in, and you'd write 3 above, and you're done. So 122. Number 122. In fruit flies, the allele for vestibular wings is recessive to the allele for wild type wings. So let's make the vestibular wing is recessive, make it little v, and the wild type is dominant, so that's big V. And the allele for white eyes is recessive, and allele for red eyes, so big R for the red and then little r for the white. Now, key to the whole thing here is that the gene controlling the wing type is on an autosome. This is a chromosome that's not an X or Y chromosome or a sex chromosome. So the wing is basic autosomal line of inheritance, whereas the gene controlling eye color is carried on the X chromosome. This is an X-linked gene. Okay, it's important. So we're going to have to do a cross here, a true breeding female. So let's draw our female, X and X, 2 X's for a female. And we know that the true breeding female with a wild type wings. So it's going to be another chromosome, and it's going to be a chromosome that it's wild type wings. That's going to be the dominant. So both of these, okay, are going to be a big V. So we have 2 different chromosomes here, but they're wild type. And of course, the X-linked gene is the eye color, and they said with white eye. So a true breeding female with wild type wings. If it's true breeding, it's going to be homozygous dominant, and it's crossed with a male. I'm sorry, with wild type wings and white eyes. So the white eyes are recessive. So they're held on the X chromosome. So I'm put a little R here. So because homozygous recessive, this is going to give us the broken gene, which is the white eyes, and the other chromosome is in this gamete is going to be the big V for the dominant. And true breeding means that they're the same for both. Now we're going to cross this, okay, with a male. And a male is X and Y. These are the two different alleles here. Now they said the male with a vestibular wings. Now vestibular wings is recessive, so there's going to be a little V here for both. That's the only kind of chromosome you can get because they're homozygous recessive. And with red eyes. Now this R only goes, big R only goes on the X because it's not carried on the Y because it's an X-linked gene, as we've seen before. So let's do the cross and do the Punnett square to see what we're going to get in terms of phenotypes, assuming there's no what? Linkage, okay, of these alleles. And there probably isn't because they're in both two different places, two different chromosomes. So we're going to have big V, little v. This means we're going to have a homozygous, or we're going to have a heterozygote here, and then we're going to have X, okay, little r, and then X with a big R, okay, and then we're going to have a big V, little v, then we're going to have the X with the little r, I should draw it like that, and then the Y, and then we'll have the big V, little v, we're doing this cross here with this chromosome, and then we're going to have the X with the little r, that should be a little r, and then they're going to drop this capital R on here, the dominant, and then we're going to have big V, little v, and then we're going to put this X here with the lowercase r to represent the recessive allele, and then we're going to have drop the Y. So the question is, what proportion of the offspring are expected to be males with wild type wings? So let's look at our males, X, Y. Wild type wings would mean that we're going to have at least a heterozygote having one big V since it's dominant. And we have that for both. So both males are going to have wild type wings so far. Okay, and then white eyes. Well, to be white eyes, all you need to have is on one of your X's, you only have one X to be recessive. These are both recessive, and they were brought to you or given to you by your mother both times. Remember, so this are both going to be white eyes because they have the one X gene with the R. The Y doesn't carry that gene, so it can't carry the dominant to mask it or to make it a heterozygote, so this stays recessive, part of the characteristic of an X-linked gene, and it's why males are easily show the phenotype because they only have one of the chromosomes, like color blindness. In any case, we can see both of these, or if we change the color, we can see that we're going to have two males, or we're going to have 50% of the offspring, okay, what proportion of the offspring are to be males and, okay, or the males, I'm sorry, it doesn't say and, with wild type wings and white eyes. So out of the 4 different types of phenotypes we have here, 2 out of the 4 are going to be males with wild type wings because the dominant trait is overshadowing the recessive allele for vestibular, and white eyes because we have the recessive on the X-linked gene, and they both have the same phenotype. So 50% of the offspring, or 1/2, would be what you want to code in. So you can do 1, code that. You can do the little slash, code that in, and 2, or you also can code in 0.5 as well. Either one will work. Okay, so number 123. Okay, number 123. ABO blood type is in humans is determined by 3 alleles of a single gene. And then basically we've talked about this. This gives us our blood typing, and here are the phenotypes, and here are the genotypes, and here are the frequencies. And then they give you a new Hardy-Weinberg formula, okay, assuming that we are in equilibrium, to figure out the percentage of the alleles based on frequency. So this gets complicated because now we have 3 different letters here when we're used to having 2. So what they want to do here is they want you to figure out what is the frequency of the allele for I, which is the recessive allele. When you have 2 of them, you get your type O, and the reason why you have 2 broken genes, you don't make a receptor, a glycoprotein. And because you don't have that receptor there, or that sugar sticking out, okay, the other 3 types of blood type of people would not consider that to be an antigen because they don't produce a certain receptor. Any case, the bottom line is, which one is it? Well, we know it's a what? A homozygote, just like that. So type O is the only what? Is the only homozygote. So now, yes, it's the only one that's, you can just get, it's the only phenotype, okay, that only has one genotype. Now you do have type AB that only has one genotype, but you don't know because it's 2PQ, 2PR, or 2QR, so you don't know which one. But type O, the recessive, the homozygous recessive, there's only one type, okay, that we can do from this formula. So whether you want to call it P squared, Q squared, or R squared, it's definitely to find the frequency of that allele. I'm going to take the frequency of the individuals that are type O, and because those individuals can only be II, which is either going to be P squared, Q squared, or R squared, who care what it is, you take the square root of 0.45. And when you do so, you get 0.67. And why am I taking the square root? Because these are individuals. So if I'm going to call this, let's say, R squared, okay, normally it's Q squared for the recessive. This is the frequency of the people in the what? Of the people in the population that have this blood type. But they want the frequency of just the allele in the population. So to go from R squared to just R, we had to do what? Square root it. Okay, and that's why we're square rooting 45. That's all that question was. Take the square root of 0.45 and you get 0.67. Now, if you want to think with me, which one is it, because you just are a person that likes to know exactly what you're dealing with, look at the patterns. The patterns are that I have 2 PQ, 2 PR, and 2 QR. Notice the R is something you have for 2 out of the 3 heterozygotes. Look at the heterozygote here. Which one is there the same allele? Look at that. There's there's the I here and the I here. So 2 out of the 3 heterozygotes, and these are the heterozygotes individual. Notice there's 2 different type of alleles. Notice the R is in 2 of them. And that R must, the one that's in 2 of them must be the I. So really it's the R squared that was, but it doesn't matter because it had to be something that was a homozygote, okay? And so you take the square root to get one of them, but if you had to know, it was R squared. And the reason we can do this for the recessive, the jumping off point in Hardy-Weinberg, is their homozygous recessive, because it's the only genotype that produces only the certain phenotype, whereas the other ones have multiples. Now this one only has 1, 2, but you didn't know who P or Q was, so you didn't know where to go there. So in any case, it only was the square root of 0.45 to find what the R was, which is actually the I. All right, so you're going to, now careful how they ask for this, give your answer to 2 decimal places. So this will be 0.67, and you can put the 0 in front and you would grid in those values, including the decimal point in your grid in. So that is how you do number 123. So number 124, biological communities contain a large number of species that are evenly distributed, exhibit high species diversity. This is true. A concept that encompasses both the species richness, which is the number of different species, and the relative abundance, the number of individuals of each species. So the abundance and the richness is how we measure species diversity. That's important. That does come up. One measure of species diversity is the Simpson's Index of Diversity. Of course, the Simpson's Index of Diversity, which is represented by the following mathematical equation. So they're giving you this equation. They're telling you what these things stand for. And based on the information below, they want you to calculate the value. So this is basically a plug and chug. Okay, although I really not liking the question. There's really nothing about biology here. So they want you to calculate the Simpson index for the community of trees here. and an answer is 0 between 0 and 1 to the nearest hundredth. So how do we do this? Well, we got to see, this means sum. You're going to add. So this is called a summation. So we're going to add all these individuals. What does n of little 1 mean? It means a number of individuals in each species. Oh, number of individuals in each species. So we're going to sum all of the different species. Since we have 4, we're going to make 4 sums of this. And let me just show you what that meant. So I'm going to take n equals 1, which is the number of individuals, which is 20. And it's going to be times the same number 20 minus 1, which would be 19. Then I'm going to add it to the next species, which is, oh, there's 34. And 34 times, because what's inside is times by that, minus 1 is 33. And you can see this is not too bad. Plus 4 minus 3, right? And minus 4 minus 1 is 3, of course. And then plus the 10 times 9 for obvious reasons. We're going to put this all over the big N, which is the total number of individuals, 68, and we're going to times that by big N minus 1, which is 67. So when we do the top, what I get is 1604, I believe. Yes. And then when I do the bottom, 68 times 67, I get 4,556. When I find this number, I get 0.352. So what do we have here, party people? This whole thing is 0.352, and it's 1 minus 0.352, and when you 1 minus that value, you get 0.647. They want you to round to the nearest hundredth. So since this 7 is bigger than 5, I go 0.65 is my answer, which I would grid in. Okay, and of course you can put 0.65 and grid that in as well. Okay, and that is what they give you, and that is the answer. They give you a range of 0.64 to 0.66 based on your rounding. But that's how we do it. And again, I don't know what this has to do with biology other than just working with some kind of mathematical equation. But again, it's a plug and chug kind of question there. All right, so number 124 was, I get 0.65 or 0.65, but they gave you the range here. All right, so number 25. Okay, number 125, the last grid in question. This really gave people some fits. A zucchini squash was peeled and cut into 6 identical cubes. After being weighed, each cube was soaked in different sucrose solution for 24 hours in an open container at a constant temperature of 21 degrees Celsius. The cubes were removed from the sucrose solutions that were different, carefully blotted to remove the excess solution, and weighed again. The percent change is due to a net gain or loss of water. And it was calculated for each tube, and the results are shown in the graph below. A straight line is drawn on the graph to estimate the results, and that's their little hint here. This is a huge hint. Straight line is drawn on the graph to estimate. Somehow we're going to use this graph, somehow. So what we have is an idea where we had different solutions, and these were the known solutions. 0 solution, okay, pure water, 0.2 molar sucrose, 0.4, 0.6, and 0.8, and these are the 4, 1, 2, 3, 4, 5 different solutions, including pure water. And using those solutions, okay, and the cubes of zucchini, we saw that there was either going to be a loss of mass. Notice the negatives right here, because why would they be a loss of mass? A loss of mass is when the concentrations were so high, there was a hypertonic. Or if we have a scenario where we have a very dilute solution, here's my cube of cucumber, the terrible cube, hey, okay, now it's better. But if we have dilute solutions, not much solute particles, and there happens to be more solute particles, let's do that in red so you can see. There's more solute particles inside, in this case, if it's very hypotonic, okay, water will rush in and the change of mass will be positive. Okay, so they say using the straight line, calculate the water potential of the zucchini squash. Well, we know water potential is equal to this would be water potential W, the solute potential plus the pressure potential. Since we have no information regarding the turgor pressure, we're just going to say in this case that the water potential is the solute potential. They did remove the skin, I guess, of the zucchini, but I'm not sure if that makes any difference there. So we're going to say the water potential equals the solute potential. The solute potential is equal to negative I C R T in Kelvin, and that's equal to the solute potential, and that's given to you, of course, in your reference table. But still, how do I do this? How do I use this? How do I use this graph? There's always an easy solution to every problem. This looks so convoluted. Do I got to figure out how much is gain or loss? Well, wait a minute. When is there going to be an isotonic? Remember, we had a scenario where we had a very concentrated solution, that's over here, and we lost mass. And then when it was a very dilute solution and a hypotonic, okay, solution, the cube's gain mass. Where was it when there was no gain or loss of mass? When was it a isotonic solution? Yes, there's no net flow. And that is the key to the entire problem. So if you look at this carefully, it's an estimate. I say this is 0.3 at this line. I say this is halfway, so I'm guessing that this line is 0.35 in terms of the molarity, which is my concentration. So see what I just did there? So I figured out that when there was zero change, now we didn't really measure that, we just used the relationship. We took it from the graph. That's why they said use the graph. We found the zero change in mass. That must be the isotonic solution, where the cube, okay, and the solution had the same concentration, so there was no net flow. That's why there was zero. And that's where the solute potential equals the water potential. And so now when you're in isotonic solution, and this is key, the water potential, okay, of your open beaker has no pressure. That's equal to the solute potential. And this potential of this is equal to the water potential, so it's all equal to the solute potential. So the potential inside the cube is equal to the water potential outside, and the water potential is just equal to the solute potential, and that's why we get rid of the pressure potential. So now we just got to calculate at 0.35. So now this I factor is how many particles it breaks apart. Well, OSes and ACEs are big chemicals that are covalently bonded. They do not break apart into ions. So that, of course, would be a 1, so -1. C is a concentration we get off the graph when it's isotonic. So therefore the solute potential is the water potential, which is the potential inside the cucumber cube. The R is a gas law constant that we'll talk about in AP chemistry next year, but we get that right off the reference table too as 0.0831. The temperature is in a Kelvin positive scale. They said at 21 degrees Celsius, we have to convert 21 degrees Celsius into a Kelvin number. So we take 21 plus 273, and this gives me a Kelvin number of 294. And then we take our handy dandy calculator and multiply that through, and what we get is a negative 8.6. All right, and that's in bars, but they asked for the question to 1 decimal place, so I rounded to that, and this answer gave you a range of negative 8.8 to negative 8.3. Okay, so my 8.6 kind of falls right in there. Okay, so that was the range, and that's how you did that. And I honestly looked at this a few times and scratched my head until I realized I needed the isotonic. So that was a tricky one, a lot going on there for one problem. And you have to understand that it's almost impossible for you guys to get to all of these questions. If you're getting to most of them and only a few you're guessing, you're doing a fantastic job. Some of these really take some time and effort to see them right away. So hopefully that helped and you utilize this resource. So good luck.